hell, where am I? Just a second. Okay. Yes, I am. So, hello Trisha, hello Manoj, hello Kishan Gowda and Minaz. Very good morning, Vijay, Aspia and Uday Charan. Ranjini has a question that, ma'am, if at all we solve mock test paper, can we really score 60 on 60 for even physics and chemistry? So, the thing is, you have to regularly give mock tests, okay? Not just this mock test, but you would have to go forth and give more mock tests, Baba, because this is very, very, very important, okay? Mock test gives you an idea about how you will be exactly doing on the day of examination as well. That is why it is known as mock test. And you have to make sure whatever mock test that you're taking up, it has to be of KCET level itself. Yeah? Ma'am, time is up. Please come to the class. Baba, I am in the class now. Okay? Hello Pooja and hello Kavita. Good morning Anjali. Vishwant, Bhavish, Bhumi. I'm a KC 30 student. Hello Bhumi. Okay. So perfect. So I think everyone is over here. Ma'am, you have done video on question paper. Will question come from that? Kavita, the thing is, like, we all suggest that you have to do previous year questions, not because the questions would be coming exactly like that, but because the concepts are repeated. Okay, the concepts are mostly repeated, but all you have seen me solving the questions in 15 days challenge. I've shown you so many concepts which have been repeated from the same chapter itself. So you get an idea of that, and probably when you solve more questions on the same concept, obviously that concept becomes what's cemented in your brain, and you would be able to solve any question related to that, right? So that is the reason. Sharon, a very, very good morning, good happy soul, and Mola, good morning. So my dear children, I think we can go ahead, and I think we are ready. So hope everyone is ready for the full length test of biology. Make sure you give me your answers, and let's go ahead with the first question that is there. Question number 1. The number of individuals of population who left the habitat and gone elsewhere during the time period under consideration is known as, what do you call this process? Where there are a group of organisms over here, and basically they have left this group and went elsewhere. What do you call this? Natality, mortality, emigration, or immigration? And you have to give me your answers like 1, A, B, C, D. So please give me the number 1 and then give me the answer, because then only I'll get to understand which question you're answering, right? So everyone is saying C. Pavitra says C. Okay, Kavita, Trisha, and Ananya, Happy Soul, Bhavish, Rishika, Kishan, everyone. Chandana, exit. So everyone is saying C, Afan, and yes. So the answer for this question is actually C, emigration. What do you mean by emigration? Emigration means, for example, I have lots of people, you, me, Chandana, Bhavish, and so many others. Kishan, lot of people are over here, and one day Chandana feels that I should not be here, I should be moving out. This process is known as emigration, and probably that person would be going somewhere else. Yeah, E for exit, always remember that. Now one more question to all of you guys. Sandy, good morning, Macha. So one more question. Emigration will lead to decrease in population density of a population or increase in population density of a population? Emigration, which is there, does it decrease the population density or does it increase the population density? Trisha says it decreases. Yes, definitely it decreases. Remember, there are 2 factors among them which decreases the population density. One is mortality and one is emigration. Mortality, because mortality means the rate of organisms who are dying. So if people are dying over here, Baba, obviously population density is going to decrease, and also emigration leads to decrease. Perfect. Chandana as well as Sangeeta, Bhumi, Alina. Hello, Alina. Rishika, great. So the answer for the first question was pretty easy, right? Ilu Khan, perfect. Good morning, Noha. Let's go ahead with the next question. Question number 2. The rate of biomass. Now here you have to make sure that you give the answers properly, read the question properly first of all. Okay, hi ma'am, big fan. Oh my god. Okay, so the rate of biomass production and the rate of production of organic matter during photosynthesis are called respectively as what? So rate of biomass production is known as what? You have to choose from here. Then rate of production of organic matter during photosynthesis specifically is known as what? That is the question. What do you think is the answer for this? Okay, 2B. Listen, you have to make sure that you are reading the question carefully. Okay, so Vishwan says it is B, Tanu says it is B, Kishan says it is B, Chandana says it is B, Ilu Khan also says it is B. What if I tell you that you have to think beyond? Probably the answer would not be B. Okay, if at all I'm asking you to think beyond. Think beyond. Sriyash, Narendra, hello dear. Alina, I'm really good. Thank you for asking, Macha. Okay, Kavita says the answer for this question is D, and I think before that Vijay was the first person to give me the answer as D. Yes. Some people are still confused. Yeah. So Pavitra also says, so the answer for this particular question is actually D. Listen to me very carefully. Why not all of it? There is nothing such as total productivity given in your NCERT. Yeah, there is nothing such as total productivity given in your NCERT, which makes sure that the first A1 is not definitely the answer. So A1 is ruled out. Now use elimination over here. GPP, gross primary productivity. Can I apply rate of biomass production? I am not mentioning where. Rate of biomass production as in by the plants or anything. Am I mentioning that? No. So obviously the first one cannot be GPP also, because GPP is applied when I am talking about plants in general, when I am talking about photosynthesis and everything, the production of organic matter after photosynthesis and things like that, right? So GPP is also ruled out. NPP is basically the amount of thing that is available for the next group of organism. That also does not apply over here. So what is left? We are left with productivity and GPP. This is the answer. D is the answer for this particular question. Listen to me if at all you still didn't understand. Listen, rate of biomass production. I'm not mentioning who is producing this or what is happening over here. I'm just mentioning what do you call the term when you are talking about the rate of biomass production. It's known as productivity. I'm not mentioning primary, secondary, nothing. Rate of biomass production is known as productivity. Done, done. Now the rate of production of organic matter during photosynthesis. Now I'm specifying, I'm putting a term into it, that is during photosynthesis. That means photosynthesis, who are doing, Baba? Producers are the only one who are doing it. Yeah, so basically you're talking about the producers which are there. What is the rate of production of the organic matter by the producers? Producers as a whole, together, all of them. So what do you call this? Gross primary productivity. Primary productivity because they are producing it, they are primary producers, and gross because you're talking about lot of plants in general, because here there is no one plant mentioned. They're just saying production of organic matter during photosynthesis. So it can be multiple plants. So I can consider GPP out there. So the answer for this question is going to be D. Is this particular thing clear to all of you? Is this particular thing clear to you guys? My hairstyle. Okay, we'll talk about my hairstyle right now. Is this question clear to you? If it is, can you just give me a quick thumbs up? If it is clear to you, can you just give me a quick thumbs up? That is very important. Without thumbs up, I do not understand anything. Okay, so just give me thumbs up. Yes, perfect. Thank you so much. 2B, GPP is there. Baba, GPP is here, but you're talking about, listen, you have to find the term for this first and then the second term, na? So rate of biomass production is just known as productivity. You're not mentioning, okay, which organism is producing it and blah, blah, blah things. Cool? Yes. Ma'am, I can't understand. Repeat. Okay, I can repeat it. So guys, just give me a second. I'll repeat it. Okay, there are some children who are not understanding it. Cool. So, listen, you have to look the question in two ways. The first part of the question ends over here. The second part of the question starts over here. Okay, what do you call the rate of biomass production? Here, are they mentioning about the producer's name? Are they mentioning about that the cow is producing, or basically the tree is producing, or the plant is producing? No, they're just asking you, what do you call that term when any organism is basically producing something and you are basically checking the rate of the production of it? You call the term as productivity. No primary productivity, no secondary productivity, no gross, no net, just productivity is the term that you use. And over here in the first option, where do you see productivity? Only over here. Only over here you see plain productivity, which is why our answer is already found. Now the next part of the question is rate of production of organic matter during photosynthesis. Now here it has been specified that what is the production, what is the rate of production during photosynthesis? Who is doing photosynthesis, Macha? Photosynthesis is done by what? The plants. So basically you are being asked, what is the rate of production, rate of organic matter production by these plants during photosynthesis? And what do you call when I'm talking about plants in general, the primary producers, and when talking about all of them together, you call this as gross primary productivity, which is why the second part of the answer is going to be this. Okay. Yeah, perfect. Now I think it's understood. So this is something that is given in your NCERT. Please make sure you go through the text which is there. Clearly they have mentioned about all of this thing, and I could not expect a question coming off from here. Even I was very much confused when I read it for the first time. So rate of biomass production is known as productivity, plain productivity. Okay, so yes, please remember that. And ma'am, voice is little lower. Is it fine now? The voice is fine now? Yeah. Good morning. Hello, bustling appa. Perfect. Cool. Now the next question that is there, question number 3. The 3-5- phosphodiester linkage inside a polynucleotide chain serves to join what? So you have got the options over here. You have to choose from the options. Macha, come on. Come on. Let's go ahead and let's start off. Okay? Come on, come on. Go, 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 go. And just give me a second. It's somehow too hot. Let me just... Okay. Okay, I think that works. 3D, 3C, oh my god. Hi Macha, hello Harish. Vijay says the answer for this question is C, okay. Swami says C, Afan Malal says it is D, Kavya says A. What is this? You are giving me ABCD option, huh? Guys, you have to choose the option. Illu Khan says A, but most of you guys, I think you are saying it is C. It is C, right? Most of you are going to say. Guys, where are you getting these questions from? So this is a question from the ABHYAS KCET test series. Okay, this is a full-length mock test from the ABHYAS KCET test series out there. Yes. Chalo. Let's go. Since most of you guys are going with C, let's go with C only and let's check it out. But before that, when I'm talking about 3-5- phosphodiester linkage, I need to have an idea about DNA. So, Baba, DNA is made up of what? A pentose sugar. This is the pentose sugar. Why do you call it pentose? Why not mentose and things like that? Because Baba, only 5 carbons are there. I know that's a bad joke. So 1, 2, 3, 4. Where's the 5th carbon? It's outside. Okay, it's outside. Now the first carbon, basically, it is attached to a nitrogenous base. What do you call this bond as? This is known as glycosidic bond. Okay, this is known as glycosidic bond. Okay, cool, ma'am. Okay, what about the 5th carbon out there? 5th carbon is attached to a phosphate group. Okay, it is attached to a phosphate group. Now, Baba, what do you call this whole thing as when you have a nitrogenous base, a pentose sugar, a phosphate group? I will call this as what? A nucleotide. I will call this as a nucleotide. Okay, ma'am. So, if at all I want to make a DNA, I need multiple nucleotide, right? Yes, ma'am. So, what do we do? Again, make a pentose sugar over here. 1, 2, 3, 4, 5. 5th carbon is still outside. 1st carbon is attached to what? Nitrogenous base. 5th carbon is attached to phosphate. Baba, this and this cannot be staying in air. It needs some connection, right? There should be some connection. So the connection is made from here. The 3rd carbon that is present over here, Baba, attaches to this phosphate group. So both these carbons at the 5th position and the 3rd position are attached to a common phosphate right over here. And what do you call this bond as? This bond is known as phosphodiester bond. Actually, you call this bond as phosphodiester bond, the bond between the carbon and the phosphate. Here an oxygen is also there. That is why you call this ester bond. Similarly, there is one more ester bond over here. Now, since you have 2 ester bonds over here, we call this phosphodiester bond. Since phosphate is also there, you call it phosphodiester bond. Now, what is it connecting? It is connecting one nucleotide with the another nucleotide. This was a nucleotide, this is another nucleotide, which is why we say the 3-5 phosphodiester linkage is therefore connecting these 2 nucleotides. Is this particular thing clear? One DNA strand with another DNA strand? No, Baba. One DNA strand with another DNA strand, you get nitrogenous bases in between. You do not need these guys in between. Now, one nucleoside with another nucleoside, what do you mean by nucleoside? Nucleoside means if there is no phosphate over here, it becomes a nucleoside. Only pentose sugar as well as the nitrogenous base. Arey Baba, you need phosphate, otherwise who will connect? So definitely not this. This is the answer. One nitrogenous base with the pentose sugar, again, that is known as glycosidic linkage. Why are you including all this thing? So if at all this particular thing is clear to everyone out there, just give me a quick thumbs up. Come on. Just give me a quick thumbs up. One thumbs up, everyone. So I'm just solving one mock test salinity, not all of them. Okay, one mock test is what I'm solving over here. So make sure you listen to me carefully, so that you will also get an idea of how a mock test work and things like that, and you can basically solve a mock test for free, right? That is the logic behind this. Yeah. Oh, okay. Perfect. Ma'am, poll. Baba, poll is not there. Bhumika, you want to ask me something? Please ask. And thank you for giving me thumbs up. And my dear children, I was saying something. What was I saying? Yes, so I'm so sorry for not reading out your names. I know that you guys were answering questions. Yeah, the reason I was not reading out is that I'll read out in between. Why? Because I might be taking too much of time reading your names. I got a complaint like that in the comment section that, ma'am, you take too many names. Is that a problem? I think yes, for the time consumption it is a problem, but yeah. Hiya. Okay. Thank you, creative. Okay. Ma'am, all KC30 classes are in YouTube, ma'am. Which classes are there in YouTube, dear? The PYQs are still there itself. The DPPs are still there. Okay. Mohana Kumari, can you please say, can we take phone and keep it outside the center? It will be very risky. You do not know what center you are getting, right? It's better to, if at all you have your parent or someone accompanying you, it's okay. Otherwise, not recommended. Okay? Okay, Darshan, thank you so much. Okay, let's go ahead with the next question out there. Question number 4. Assertion: in eukaryotes, both introns and exons are transcribed to form hnRNA. The reason: splicing is required in prokaryotes. What do you think is the answer for this particular question? Read out the question carefully and then only you need to go ahead. Ma'am, I think you need fan. Yes, definitely I need fan. Okay, so what do you think is the answer for this question? Macha, come on, da. Okay, Trisha says the answer for this question is D. Manjula, it's definitely not enough. You have to keep on practicing. Okay, Nisha says it is C. Kavya says it is D. 4C. Okay, Kavita says it is C. Mohana says A. Kenchappa is also there. Hello. And Tarun is also there. Okay. Nisha, Tarun, everyone says C. Bhavish has a different answer. Okay. Solinity says D. Okay. M3Y says C. Ilu Khan. Okay. Sangeeta, Sandy, and Mahadev, Dimonish. Okay. So let's see. Animation. So the answer for this particular question, I have to check. In eukaryotes, both introns and exons are transcribed to form hnRNA. Yes. So basically, this is the DNA. The DNA have some non-coding regions. You call them as introns. They have some coding regions. You call them as what? Exons. And as such, it's like rectification, copy to copy. Some of you might have tried copying in the paper exam, right? So we also try copying. What exactly happens? Sometimes we think that we will get the exact answer, but we actually do not copy the exact answer. So anyways, this is your DNA, and over here we have the RNA that is being made. Now this RNA that is being made is known as what? hnRNA. So whatever is there is over here also in the case of eukaryotes. Okay. Now splicing is required in the case of prokaryotes. Now in the case of prokaryotes, Baba, in the DNA they do not have introns and exons. They just have exons. Only the coding regions are there. Only exons are there. So do you think that you need to remove the introns? Or if there is no intron, what will you remove? Okay, what will you remove? You have nothing to remove. So this particular statement will be wrong. Splicing is required in prokaryotes. No. Splicing is required over here. So listen, exon came over here, intron also came over here. Exon came over here, intron also came over here. Now when you make an mRNA, you have to make sure that there are no introns. There are only exons. Why? Because this mRNA is going to go outside the nucleus, undergoing translation. At that particular point of time, if it takes introns also, the ribosome will get very angry and say, why did you bring introns? I do not need it for production of proteins. So you have to remove the introns before only. So while you make the mRNA, there is one process known as splicing, where you basically remove the introns out there. This is a feature of eukaryotes, not prokaryotes. So I'll say assertion is true, but the reason would be false. Assertion is true, but the reason is false. The answer for this question is C. Okay, heterogeneous nuclear RNA. Okay, heterogeneous nuclear RNA. hnRNA is heterogeneous nuclear RNA. Cool, macha. For everyone who gave me the answer as B and D, I hope it's clear now. It's clear? If it's clear, quickly give me a thumbs up. Quickly give me a quick thumbs up. Ma'am, can you explain assertion again? Yes, I can. In eukaryotes, both introns and exons are transcribed to form hnRNA. As in DNA which is there, as a whole it is getting transcribed. That is what you mean. In the DNA, in the case of eukaryotes, you have the exons and you have the introns. As such, whatever information is there is being transcribed to hnRNA. That is correct, right? That is correct. Later, these introns are removed when you make an mRNA. Okay? I hope that works. Okay. Ma'am, I have logically a question. Why prokaryotes don't have split genes? Papa, they are very primitive organisms. They do not have mechanism to cut off all these things. That is why probably they do not have all this, what do you call, introns and stuff like that. Yeah? Yes. thumbs up wala emoji. Okay, perfect. So going ahead with question number 5, Macha. How are transformants selected from non-transformants? Okay, transformants selected from non-transformants. Presence of more than one recognition site in the vector DNA. Okay. Presence of alien DNA in the vector DNA results onto insertional inactivation of the selectable marker. Antibiotic resistant gene gets inactivated due to insertion of alien DNA, both B and C. What do you think is the answer for this question? Thank you so much, animation. Your style is so nice. Ma'am, this video is enough. No bustling up. You have to practice more questions, Macha. Okay. Kishan says D is the answer. Kavita says D. Sandy also says D. Farah says D. Darshan and Noha and Harish, Swami, Ilukhan, Hitesh, Rohit, Tarun, Kavya, Kenchappa. Oh my God. Ramya, Vijay, GS Suni, Noha, Kazi, everyone, Sangeeta. Okay, everyone says the answer is D, which is why I'd be locking up D. So whenever you get an option like both B and C, most probable chances are there that could be correct. But anyway, let's see the explanation to it. So what do you mean by transformants and non-transformants? Basic understanding, Baba. So over here, suppose a particular cell is there, okay, and this cell usually does not have a plasmid. This cell usually does not have a plasmid, generally, okay. Now what do? I put a plasmid over here with my gene of interest, with my DNA of interest. What do? Basically, I let the plasmid get inside with the help of some methods, I let it go inside the cell out there. Now, some cell will not take it up and some cell is going to take it up. The one that is new now has transformed. I will call this as what? Ayo, where is it? I will call this as the transformant. I will call this as the transformant. And the one that has not taken up is just like before. I will call this as what? The non-transformant. Okay, so basic understanding of transformant and non-transformant is clear. I hope so. Yeah. Now we get into the next thing. That is these options which are there. Baba, when you talk about a plasmid, suppose you're talking about PBR322. You know the PBR322 has two resistant gene. One is ampicillin resistant gene. That means if at all they have this segment of gene, the cell can be grown in a medium that is having ampicillin. Ampicillin is an antibiotic that kills the the organism, the bacteria out there. But if at all a bacteria has a plasmid that has this particular gene, it provides resistance to that. It can still grow because it has the resistant gene. Similarly, you have a tetracycline resistant gene over here. Now here, why B and C is correct is that you are being asked how are transformants selected from non-transformants. So basically, you have to make sure that you have to find a way to select it. How do I find a way to select whether this is a transformant and whether this is not? Basically, I treat both of them with ampicillin. This one will not be able to grow. Why? Because it does not have the gene for it. But this one will be able to grow. Why? Because it has the gene for it. So I'll say that this is transformant. So basically what is happening over here, presence of alien DNA in the vector DNA results into insertional inactivation of the selectable marker. So over here, if at all I am putting my gene of interest in the tetracycline resistant place, what is going to happen? Something else is being put over here. This guy is going to get inactivated. This guy gets inactivated. Means that normally a cell that has the plasmid is going to grow, but if at all it has my gene of interest, it is not going to grow. It is not going to grow. Why? Because I have put something in the selectable marker area which makes this particular thing insertionally inactivated. It inactivated. Now antibiotic resistant gene gets inactivated. So it can be antibiotic resistant gene, Baba. It can be something else. In the lag Z operon, you saw that it was lag Z gene which was getting inactivated. Over here you are seeing the tetracycline. This is an antibiotic that is getting inactivated. So both the statements are going to be correct, which is why both B and C would be the answer for this particular question. Is this thing clear to you? If it is clear and we can go ahead with the next question, can you quickly give me a thumbs up? Can you quickly give me a thumbs up? Yes, perfect. Ma'am, why only these chapter questions are coming? These chapter, we have solved multiple chapters question, no? Okay. Now question number 6. A dicotyledonous plant forms a crown gall when? A dicotyledonous plant, okay, forms a crown gall when? So basically, when does it form the crown gall tumor? You might have heard about that particular disease, right? So when does a dicot plant, okay, get the crown gall disease? When agrobacterium tumefaciens come in contact with the plant, when agrobacterium rhizogens come in contact with the plant, a specific part of the DNA from the TI plasmid gene gets integrated with the plant chromosome, or a specific part of the DNA from the RI plasmid gets integrated with the plant chromosome. What do you think is the answer for that? Hema, I will be repeating it, okay? Just give me a quick second. Okay. Tarun says the answer for this question is C. Swami says it is A. Sandy says B. Vijay says A. God Gamer. God Gamer says it is C. Okay. Mahadevi says B. Bacha, what is this? Everyone is giving me A, B, C, D option. No. Give me one option. One answer. There's only one answer for this question. Check out. Okay. So initially it was A, B. Okay, Chetan, Nisha and Bharati has given me correct answer, and also the children who were at the top. Okay, so I think I should be going ahead and giving the answer for this particular question. Listen to me carefully. Over here, I have agrobacterium tumefaciens. Here, right over here, this is agrobacterium tumefaciens. Okay, why I have made this? Because this guy causes tumor in the case of plants. How does it cause tumor? It has a very big plasmid out there. What do you call this plasmid as? TI plasmid. Okay, TI plasmid. TI stands for what? Tumor inducing plasmid. It induces tumor. How? Let me tell you. So this tumor inducing plasmid has a region over here. What do you call this region as? tDNA. It is known as tDNA, or you also call this as transfer DNA. What basically happens is that when this organism is going to affect a pavam plant over here, it transfers the plasmid, and from the plasmid, what is going to get transferred? The tDNA segment is going to get transferred into the plant. This tDNA segment is the one that causes tumor. It causes tumor. So the question is, a dicot plant gets crown gall disease when agrobacterium tumefaciens come in contact with the plant. Contact is not enough. You have to infect also. This is why this is wrong. Agrobacterium rhizogens is a whole different species. That is why it is wrong. Agrobacterium tumefaciens is the only species that causes crown gall. The next thing, a specific part of the DNA, okay, from the TI plasmid gene gets integrated into the plant chromosome. That's when they get the disease. Exactly. What do you call that specific part? Transfer DNA is that specific part. So this becomes your answer. Why not this? The other option, a specific part of the DNA from the RI plasmid. RI plasmid is not there, Baba, in our NCERT itself. So how can it be RI plasmid? So the only answer for this question becomes what, Baba? It becomes C. Is this thing clear to you? Is this thing clear to you, everyone? Sri Ganesh, yes dear. Okay, Lekhna, and yes, perfect. Sandy, perfect. Harish, also perfect. A is also possible now. Kavita, why is A is not possible? Because over here, Ayyo. Your teacher might die. Okay, so why A is not possible is because, listen, over here, an organism is there. Okay, oh, so a tiger is coming. Okay, Jasmine Ma'am is right over here. A tiger came near me. Did it kill me? No. For killing me, it has to eat me, right? So it's just saying that agrobacterium tumefaciens come in contact with the plant. It has not mentioned anything about how it's working, right? So for that, you have to look at the C option. It's been elaborately been mentioned that this is how it undergoes. It makes the plant, get the crown gall disease. So obviously then the option would be C, right? Yes. Machi, explain once more. Everyone needs an explanation once more for this particular question. Is it fine? I can just explain once more. Listen to me carefully. This is agrobacterium tumefaciens. This is the plasmid that it has. The plasmid is known as tumor-inducing plasmid or TI plasmid. Why tumor-inducing plasmid? Because it has a segment of DNA that is known as tDNA, transfer DNA. So when this particular organism infects a plant, it transfers the part, that is tDNA part, into the plant and causes the disease. That is the reason. And with that, I hope now it's clear. Clear now. Guys, can I just quickly give me one more thumbs up, because I saw just 5 thumbs up out there. Not even 5. Only 3 people gave me thumbs up. Please give me thumbs up if it is clear. If we can move ahead. Okay, thank you. Perfect. Cool. Let's go ahead with the next question. During the processing of pro-hormone insulin into the mature insulin, what happens? The pro-hormone is there, that means the inactive version of the hormone, and you have to make it what? The mature form. So what do you do? What is the processing that is undergoing? Baslingappa man, crown gall means it's basically a tumor, crown gall disorder. Okay, it's basically a tumor that is being formed in the case of plants. Okay, now Navneet says the answer is B. Okay, and what about others? Kishan says B, Trisha says B, Vijay says B, and it's me, Zainab also says B, Tarun and Vijay and Bharti, Navneet, Rohit, GS Suni, Farah, RG Patil, Ilu Khan, Bhavish. Perfect. Lekhna, Vidya, Afan. Okay, so most of you guys are saying B, which is why I assume it is B only. So it is saying pro-insulin. Always listen, Baba, pro-insulin has A, you know, A, B, C, D, right? So A and B is there, but the problem is that the C comes in between. Okay, C comes in between. We say na kebab mein handi. So it's just like that. C is in between. Actually, we do not need C. So what exactly happens is that this is pro-insulin. That means the immature form of insulin. When you make a mature form of insulin, you cut this off and you just have A and B peptide, and this A and B peptide are joined with the help of disulfide bonds. So C peptide is removed from the pro-insulin. That is how you get the mature insulin. The answer would be correct. The answer is B for this particular question. Is it okay? Is it clear? If yes, just give me a quick thumbs up. A quick thumbs up. Abdul Jalil, can you please refresh and check the settings of your video, Baba? I don't think it's blurred because I can clearly see it in my phone, right? Yeah, perfect. Thank you. Okay, cool. Ma'am, fast ma'am. Dear, if at all I go faster than this, probably I'll die. Okay, I need to breathe also in between, no? So amplification of a gene of interest by using DNA polymerase during PCR may go up to. So if at all we are giving a small segment of DNA and we are asking the PCR to amplify it, how many copies can be made? Come on. How many copies can be made? Navneet says C is the answer. Amlu says C, and Cheetah is also there. Oh my God, Cheetah says it is B, and Sandy and Zainab and Amalu and GS Suni, Vidya, Rudra is also there. Sampreet is also there. Guys, if at all I'm missing out your name, I'm so sorry. Sonakshi, I'm trying to read out in between. Megha. Okay, so most of you guys are saying the answer for this question is C. C is what? 1 billion copies. Do not go and write 1 million and come back. Okay, always remember it is 1 billion copies that is being made over here. So 1 billion copies is the answer. I think that was a very easy question, which is why I'll go with the next question without asking for a thumbs up. Question number 9. Match the following. You have to match the following. Whenever match the following question comes, the rule that is there, remember the rule, guys. The rule is what? Match this one and match this one first, and before doing all of that, read all the options. So predation, commensalism, parasitism, competition is mentioned over here. Cuscuta, balanas and cactus and orchid is mentioned over here. I can give you another way around also. Some things might be there which you clearly know. For example, Cuscuta, you know Cuscuta is a plant parasite. It looks yellow in color. We have studied from 6th grade onwards. So basically Cuscuta goes with parasitism. A goes with 3. A goes with 3. Do I have an option like that? I just have one option, and Macha, I got the answer itself. The answer for this question is going to be A. Who is saying? Bharti, I can't understand here. Which question you're not able to understand here? Tell me. Baba, in NEET, you have to go in depth. In KCET, it's not like that, okay? KCET is comparatively very easier in comparison to NEET. So, yes, everyone says the answer for this question is A. Hello, Sampreet. Very good morning, dear. So, the answer is A itself. I'll tell you why. If at all, as I said, you can go by this particular trick where you spot the easy one out and you basically tag it up. So, you got Cuscuta, you know it's parasitism. Otherwise, if not, then what do you have to do? Match A first. Predation over here. Predation over here goes with what? Cactus and the moth. Cuscuta and hedge plants, basically they show parasitism. Balanus and thalamus, they show competition. Cactus and the moth, there is cactus and there is a moth that eats the cactus and keeps it in control. So that is predation. Okay, commensalism basically means what? One organism gets the benefit, another organism is neither getting benefited nor getting harmed. That is seen in the case of orchid and the mango. But anyways, competition is this next thing that you have to look forward for. Competition is balanus and thalamus. These guys are competing, and if at all you can remember, can you tell me, when balanus and thalamus are competing, who wins the race? When balanus and thalamus are competing, Who wins the race? Who is the first position? Who is in the second position? Any idea? Please tell me that also. Ma'am, can I get your number? Ayyo, Baba, I'm so sorry. Okay, Balanus is the one that is going to be occupying the first position. Always remember that, okay? When these two guys are competing, Balanus is the one that outcompetes it. Okay, now commensalism, basically as I said, orchid and the mango. Why orchid and the mango? So mango plant is right over here. It's doing its job, okay? Now the orchid said, Macha, can you give me some space? Da, I will not harm you at all. I just want to be here. Why I want to be here? Because if at all I am lower down, I will not get enough sun rays. I just want to have sun rays. That is why can you accommodate me over here. So is it harming the plant? No. Is orchid getting the benefit? Yes, it is getting the sunlight and things like that. That is why it is known as commensalism. Parasitism means one organism would be a host, another organism will be a parasite. So here Cuscuta is the plant parasite and plant is the host out there, right? So the answer for this particular question is going to be A. If this question is clear to all, Macha, give me a quick thumbs up. Mahadevi, what are you not understanding? Listen, please answer my question. Swami, Swami. 8th question and why option A. Okay, 8th question. I'll come back to it. Okay, I'll come back to it. Shrishti, your teaching is very beautiful. Thank you and understanding very much. Thank you so much. Okay, so guys, since you have given me thumbs up, I understand that you have understood. But one child has a doubt in question number 8. If at all I can, just solve it up. Just give me a second, okay? Amplification of gene of interest by using DNA polymerase. So this is a very straight question, okay? So in that, PCR, polymerase chain reaction, is where you give a segment of DNA. And basically, it can make multiple copies out of it. So it has just been asked, how many copies can it be made? So the straight answer would be 1 billion copies. Okay, 1 billion copies can be made using PCR. It's as simple as that. Okay, perfect. Okay, question number 10. Barnacles growing on the back of the whale is an example of. Now I'm excited for this question. Why? Because no, I have brought a picture of barnacle as well as the whale. So barnacle, which is there on the back of the whale, is an example of what? Oh my God, you have given me the answer. Darshan says it is B. Zaina was the first one to say it is B. Sampreet and Hitesh and Rudra, Komari and Trisha, Asif, Sandy and Darshan, Noha, Amalu, Bhavish, Bharti, Rohit and GS Suni, Kavya. Okay, so Vinod, Muhammad Atif, hello dear Tarun. So all of you are saying, most of you guys are saying the answer for this question is going. Chandana, Sonakshi is saying the answer for this is commensalism. What do you mean by commensalism? Again, one organism is getting benefited, another organism which is there, which is neither getting benefited nor harmed. So if at all you have never seen barnacle and a whale, tan, ta -dan, this is how it looks. So this is the whale out there. On the top of it, you can see these small chintu guys out there. These chunnu guys that are there, they are known as basically the barnacles. And what do they want? They want this transportation. They are not going to harm the whale, okay? They're not going to harm the whale. They're not benefiting the whale. They just want to transport from one location to another location. So when you were kids, you might have tried this hopping onto your parents and say elephant, hathi games. The same thing is happening over here. They just want transportation. So this guy is getting benefit of it. That is the barnacle. Okay, clear, Macha? Is it clear, the question? If yes, give me thumbs up. Ding, ding, ding. Good morning, Clara. Very good morning. Okay. Kishan Baba. Okay. Perfect. Thank you so much. Let's go with question number 11. And it goes like this. A person admitted to hospital as he had myocardial infarction. A cardiologist injected him streptokinase. Why? Someone was having myocardial infarction and the cardiologist says, I have to inject streptokinase. Why? What is the reason for streptokinase? Love you, ma'am. Thank you so much. Love you too, Queen. Okay. So the answer, okay. Rudra says C. Okay. Sampreet says X. X is there an option? No X is there. Okay. So Akshita says C. Okay. Kishan, Sampreet now says C. Kishan, and it's me, Zainab, Kavya, Trisha, Chetan, Bhagya, GS Suni, and Jayanti. Hello, Rohit, Noha, Kazi, Shivakumar, Fatima. I'm just reading in between, okay? So most of you guys are saying the answer for this question is C. If someone is asking my name, can anyone tell what's my name? So Baba, why do you inject streptokinase? Because streptokinase is a clot buster. That means suppose over here you have the blood vessel, right? So blood should be passing from this blood vessel, but what happens suppose over here you have you have some cholesterol, you have some fatty things and everything being accumulated over here. What exactly happens when all these guys accumulate? Our poorer blood cells which are there, they are not able to get transported. They will not be able to get transported, which is why you have to make sure there is someone who is going to burst open the clots out there, and basically your red blood cells can easily move through. So that is clot buster, which is the streptokinase. It's not because it stimulates the heartbeat, it's not because it reduces hypertension, it's not because it reduces the level of the blood cholesterol. And also one more very important question to you guys, and that is, thank you for telling me the name. Okay, one more very important question for you guys. Who produces streptokinase, Baba? Can you tell me who produces streptokinase? Yes. Thank you so much. Hina and Mohan, everyone helped. My name is Jasmine. Okay. Guys, who produces streptokinase? Everyone who is listening to me. Streptokinase is produced by Streptococcus. Exactly, Trisha, Zainab, as well as Bhavish, as well as Sumit, everyone, Sampreet, Yashwant, Tarun. And Streptococcus is a bacteria or is it a fungi? Streptococcus which is there, is it a bacteria or a fungi? Love you too, Chetan. Okay, tell me that also. Guys, the reason I go ahead with all this thing is that I tell you that questions are not repeated, concepts are, you have to make sure your concepts are super strong. Yes, it's a bacteria. For those of you who are saying it's a fungi, guys, it's time to rectify. Streptococcus, coccus term comes, no, just understand it is a bacteria, not a fungi. Okay, and please be sure about all the things, streptokinase, your cyclosporin A, and everything, statin, everything is super important. Okay, let's go. Now, question number 12. The bigger cell of a pollen grain that contains abundant food reserve and a large irregularly shaped nucleus is what? The bigger cell of a pollen grain that contains abundant food reserve and a large irregularly shaped nucleus is what? Okay, so most of you are saying it is B. Just give me a second. B, vegetative cell, B. Okay, Kushi Karan is also over here. Hello, dear. Okay, so the answer for this particular question is going to be B, vegetative cell itself. But those of you who are confused, listen to me carefully. This is your pollen grain, and there is a very big cell. I always tell you, very big cell is vegetative cell. So you can remember, very big cell for V. Okay, now this guy has an irregular nucleus out there. Okay, the shape of this nucleus is irregular. In the cytoplasm of this very big cell lies a very small cell. It has an elongated nucleus. What do you call this as? Generative cell. What is it generating out there? Electricity? No, Baba. It is generating what? Gametes. That means actually mitosis happened. That's how you got these two cells. Again, the generative cell undergoes mitosis to produce two male gametes. Have that logic feeded in your brain. Okay. So this is for gamete production. Now there should be someone to help it provide nourishment and everything, and that is the role of vegetative cell. It contains abundant food reserve for this particular reason itself. Yeah, and also remember, there is one more role. Okay, there is one more role of vegetative cell. Anyone can tell me, apart from nourishing this papa guy out there, there is one more role of vegetative cell. If at all you tell me that, I'll give you chocolate when I see you. Yes, Queen, I can tell. I can definitely tell you the name of the chapters as well. This is from sexual reproduction in flowering plants. Yeah. Guides the gametes. Does it guide the gamete? I am asking you one question. Apart from what do you call providing nourishment to the generative cells to form the gametes, what is the other role of vegetative cell, guys? Any idea? Dense cytoplasm. Yes, definitely it's dense cytoplasm, but apart from that, food is providing. I'm telling you. Okay, tell me. Pollen tube, someone said that. Who is that? Trisha said pollen tube. Exactly. Listen to me. Pollen grain is there. Pollen grain is going to land on the stigma, right? Who forms the pollen tube? There has to be someone who forms, basically gives the cytoplasm and everything for that. That is vegetative cell. It is also known as tube cell for that particular reason. It helps in the formation of pollen tube. Always remember that. Okay, produces the pollen tube. Exactly, Queen and other children also. Okay, Swami, it converts tube cell. Yeah, it helps in the formation of the tube cells. Cool na? Will you remember it? This is important. As I said, Do not ratify a question. I'm telling you again, okay? And you have to listen to me carefully if you are someone who, because I have been there and I am telling you, okay? I have also done ratification of the question. This is this. I remember, okay, I'm able to solve this question. Again, when I solve this question, I'm able to remember this. No, you need to know the concept behind it. You need to solve every question that could be coming from the same concept. That is the logic. And for those of you who are asking me, will the PYQ get repeated? PYQ will not. I'm telling you, the concepts are going to be repeated. So do not do that. Yes, so this question is clear. If yes, give me a thumbs up, everyone. If yes, give me the thumbs up, everyone. Okay, in our college everyone do ratta, don't do that, okay? Question number 30. This is also from sexual reproduction. Okay, I'll tell, okay, anyways. Sporogenous tissue of microsporangia is what? Sporogenous tissue of microsporangia is what? A group of compactly arranged homogeneous cells occupy the center of microsporangium, present inside the young anther. All are correct. What do you think is the answer for this particular question? Okay, Fatima Faiza says the answer for this question is D. Okay, Rudra says it is A, Bega says it is A, Kishan says it is D, okay, Kavya says D, Sumit says D, Amlu says D, Jayanti says D, okay, Amlu D, and Atif says D, Sampreet, Jani, Irfan, okay, Sandy, it's Zainab, Trisha, Rudra, Bharti, Ramya, Nauman, hello, Bhavish, Kalpana, and everyone out there. Okay, Saimullah Khan, Shivakumar. Okay, so the answer for this question is going to be D. All of them are correct. And I'll tell you a method whenever you're not able to solve the question. Yesterday I made on a video on guessing MCQs. Have you watched that particular video? Don't watch it now. Okay, watch it after this particular session ends. So in that I am telling you that there are chances, most of the chances, whenever you get all of the above option, that could be correct. Apply this only when you're not able to solve questions. Okay, but anyway, let me talk about sporogenous tissue. Let me see if at all I have picture. I have the picture. Okay. So over here, Baba, what is there? This is your anther. You suppose this is the anther that is there. I have cut it and I'm looking from the top. When I'm looking from the top, it looks to me like this. Okay, it appears to me like this. And over here in the center, what is this? This is orange. No, this is not orange. This is what? Sporogenous tissue. And your sporogenous tissue is made up of what? Lots of similar type of cells are there. And these cells, Baba, are going to later on form the microspores. And the microspores are later going to form what? The pollen grains out there. That is the logic. So you have been asked about sporogenous tissue. Let me see what all things are correct. Group of compactly arranged homogeneous cells. Homogeneous cells means similar type of cells. Are they? Yes. Occupy the center of microsporangium. So this is a microsporangium, this is a microsporangium, this is a microsporangium, this is a microsporangium. Does it occupy the center of each of them? Yes. Present inside the young anther, Baba, this is the young anther, and over here it is present. So all of them are absolutely correct. Which is why the answer for this particular question is going to be D. No, ma'am, it's A. Why are you saying it's A? Shushti, I'm so sorry, Baba. So many, what do you call, the names are going on, which is exactly why I'm not able to. Sumit Baba, there is no polls. You have to give me answers like this. I'm so sorry. But that makes it more interactive, right? Yes, I get to interact with you more. So, yes, you look similar to my college bio teacher like Afrin ma'am, is it? Okay. So, guys, if this question is clear to all of you, can you quickly give me a thumbs up? Amalu, you look so pretty. Thank you so much. Okay, guys. I think it's clear. This is from the chapter Sexual Reproduction in Flowering Plants. Perfect. Okay. And by the way, everyone who have not hit on the like button, hit on the like button right now. It makes sure that the video reaches to entire people out there. When you click on the like, the reachability or it increases. Okay. So make sure you do that, and do not forget to hit the subscribe button as well, if at all do you think this channel is helping you. Now question number 14. The slurry left after methane production from the cattle dung is... The slurry left after methane production from the cattle dung is burnt, buried in landfills, used as fertilizer, used in civil construction. What do you think is the answer for this? Fatima says it is C. Okay. Sumit says it is C. Akshita says C. Nakshatra, yes ma'am. Okay, thank you dear. Okay, Trisha says C. Sampreet has a different answer. Darshan says C. Bhagya C. Zainab, Hitesh, Bharti Queen, and Rohit. Ranjita. Okay. Now, man, Hemant, Nakshatra. So most of you, Mad Ranger is also there, Manjanath, Hemant, okay, Hanumanta. So the answer for this particular question is going to be, see, as you guys are saying, it is used as fertilizer. What do you mean by slurry left after methane production? Listen to me carefully. Over here, this is a biogas plant, okay? You have to use your imagination. Plunk, plunk, plunk. I am going to put the cow dung over here, okay? Why cow dung? Because cow dung has methanobacterium in them. Cow dung has methanobacterium in them, okay? Now what do? I put the cow dung and basically I mix with water. So the cow dung, everything moves to this particular place. If at all you look this particular place, it is entirely closed, right? So that means it is giving an anaerobic environment. No oxygen is there over here, right? Now the methanobacterium is also there and everything is also over there. Basically the methanobacterium in anaerobic environment, what does it make? It makes the biogas. It makes the biogas. So there will be an inlet to take the biogas outside. Now whatever is left behind basically is going to be transferred over here in this particular area, and another tank out there. What is this thing? This is the leftover thing out there. So shall I throw it away? No, I can use this as a fertilizer or basically as a manure. So that is the question. The slurry which is left after the methane production is known as what, or used as what? It is used as the fertilizer. Is this particular thing clear to you? It is helpful for crops. Exactly, it is very much helpful for crops. Clear na macha? Yeah. One more question that I want to ask you. Two organizations were responsible for this biogas production. Can you tell me the names of these two organizations? Everyone. Two organizations were responsible for the biogas production and stuff like that. Can you tell me the name for it? Ma'am, clear explanation. Thank you so much. Guys, I hope you're drinking water. Please drink water. Slurry removed through other outlet and used as a fertilizer. Exactly, Aphanomelia. Okay. KVIC, someone says KVIC, exactly RG Patil, it is KVIC, Khadi Village and IARI. It's Astro is also saying IARI, exactly. So there are two organizations, KVIC and Indian Agricultural Research Institute, which is known as IARI, okay. Khadi Village Industries Commission, KVIC, and Indian Agricultural Research Institute. So these are the two organizations which were responsible for the biogas production. Please remember these things also. Okay, exactly, everyone. Sharanya, Sayamullah, Chetan, Bharti, everyone. Guys, I'm telling you again, if at all I'm missing out your names, I'm so sorry. I genuinely don't want to miss out your names, but yes, due to the time constraints. Okay, let's go ahead to the next question then. Question number 15. If the distance between the two consecutive base pairs is 0.34, and by the way, this is from the chapter Microbes in Human Welfare. Okay, so someone was asking to tell me the name of the chapters as well. So the distance between two consecutive base pairs is 0.34 nanometer, and the total number of base pairs of a DNA double helix is in a typical mammalian cell is 6.6 × 10⁹ base pairs. Then the length of DNA is approximately equal to what? Is approximately equal to what? So most of you guys are saying it is D. Is it D? SD Stragan, no, Tarun and Mad Ranger, Sharanu, Sampreet, Akshita, Karan, okay, Amlu, Kavya, Bharti, Zainab, and Sonakshi and Darshan, Vandana is also there, Hemant, RJ Patil. Everyone says the answer for this particular question is going to be D. What is D? 2.2 meters. And for those of you who are like, what is happening over here? So listen to me very carefully. Over here you have the base pairs, right? So you are saying that the distance between two consecutive base pairs is what? 0.34 nanometer. Okay, and then you are also saying one more thing, that there are 6.6 into 10⁹ base pairs out there. You are saying there are 6.6 in 10⁹ base pairs in total, and in between all of them you have a space like this. So basically what do? If at all you want to find the entire length of DNA, you multiply both of them. So the answer would be coming around 2.2 meters. The answer would be coming around 2.2 meters. So just have to do the multiplication and also make sure you change the NM to meters. That's all. Okay, so Papa, first PUC questions, everything is going to be mixed over here. This is a mock test that I have taken from ABHYAS. Okay, this is one mock test that I have taken from ABHYAS. You will find first PUC questions also over here. Okay, don't worry. Okay, next question that is there, it goes like this. Question number 16. Arrange the following. By the way, this is from the chapter Molecular Base of Inheritance. Okay, now arrange the following in a sequence of stages of microsporogenesis. Come on. Come on, arrange the following in a sequence of stages of microsporogenesis. Ma'am, some tough questions. Baba, questions can be of only KCET level, no? Yes. So, yeah. Come on, come on, come on. So, Nakshi, question number 50 now. Just a second. If at all, I'll request you, if you can just go back and listen to it once more, or can you just wait, because I think they will kill me if at all I go back, but I'll still try, okay? So, most of you guys are saying it is D, but some of you are going with C also. Why? Whenever you get this type of questions where you have to arrange the options, okay, please understand, go slow, okay? You have to go slow. So, Baba, look at these options carefully. Microspore tetrad is there, sporogenous tissue is there, release of pollen grains is there, microspore mother cell is there, and microspore dissociate from each is there. So, basically, we all know sporogenous tissue is something that is going to occupy the first position, right? Right, we all know that. So wherever you see 3 as the first one, that is the option that you have to choose from. So I can cancel these 2 options and I have these 2 options left now. Okay. Now the last thing that I know from here would be what? Release of pollen grains, because nothing else is going to be coming up. Release of pollen grains is the last event that is going to happen. Right. So this is going to the last event. So if at all the things are ending with 5, probably there are chances that it could be the answer. So 5 is this one. Okay, it is ending with 5. So my answer could be this. Let me check this option now carefully. Okay, so that saves a lot of your time. So now if at all we look at this option, first is sporogenous tissue, third one, okay. Then after that you have 2. 2 is what? 2 is microspore mother cell. Inside the sporogenous tissue, do you have microspore mother cell? Yes, you have that. Okay, then you have microspore tetrad. Okay, the microspore mother cell is going to undergo division and you are going to get microspore tetrad. Okay, at the end of it, correct. Microspore tetrad which is there, they are going to dissociate from each other. Is that true? Yes, that is also true. And the last one, release of pollen grains. So the answer for this question is going to be D. Did you understand the trick? Please go ahead with the trick. Look at the options, number it first, and then do the checking over here. That will be helping you save your time. Okay, so D, everyone who said D is the answer for that particular question, you guys are absolutely correct. Okay, Yam Kshatriya Baba, we will be coming with complete revision where we'll be doing that. Okay. Cool, perfect. So if it helped, can you quickly give me a thumbs up? By the way, children, everyone who think that it has helped you, please give me a thumbs up. By the way, by then I'll just explain the question. So someone asked me the question. Listen, it is given that between 2 base pairs, the space is what? 0.34 nanometers. And it has also been mentioned that there are how many base pairs? 6.6 in 10 to the power 9 base pairs. That many base pairs are there. So if at all, between each of these base pairs, you have 0.34 nanometer space. And if you multiply both of them, don't you think you will get the entire length? That is how you have got 2.2 meters. So you just have to multiply these 2 terms. Cool. I hope the person who had the doubt, that doubt is clear. And I hope I can go ahead with the next question. Question number 17. Okay. Hello, Jasmine Ma'am. Hello, Amlu. Okay. Next question. Oh, secondary metabolites such as nicotine, strychnine and caffeine are produced by plants for their what? By the way, this question was from sexual reproduction, and this question you guess from where. Okay, so the answer, Rakshita is saying it is D. Sampreet is saying it is B. Zainab is saying it's D. Mallikarjun is also there. Hello. Arif, hello dear. Sumit says A. Queen says D. Okay, Shalom, hello, hello, hello. Good to see you all of here. Okay, D. Yes, doubt is clear. Most of you guys are saying Tarun, Asif, Hina, Kaushal, Shreyas, Vijay, Komali, and Divya, Bharati, Rohit, Norman. Perfect. Everyone says the answer for this question is D, and D is what? Defense action. Baba, this is not from PU1, this is from your PU2 itself, and this is there in the chapter Organisms and Population, where they are telling that plants which are there, they basically produce certain secondary metabolites, and the secondary metabolites are actually used as a defense mechanism against other organisms. For example, if at all you remember Calotropis plant, okay, what does it do in acting as a defense? It produces some chemicals that can kill the organisms out there. Okay, some cardiac glycocytes. Similarly, you have other examples: nicotine, strychnine, and caffeine. So basically, they are going to help us with defense action. Okay, so always remember, this question is not from PU1, this is from PU2, organisms and population. In these sessions, do we have a break? We usually take break, but let me see with today's pace if at all we can. Okay. Vishwanayak, definitely dear, I will have to check with it, but I'll check. Okay. So if this question is clear to all of you, can you give me a quick thumbs up? A quick thumbs up. Hinak also, you will get all the different questions. Don't worry. You will get questions from all the chapter. Okay. Thumbs up, thumbs up, thumbs up. Okay, perfect. Thank you. Question number 18 goes like this. If 30 joules of energy is trapped at producer level, then how much energy will be available to the peacock as food in the following food chain? Plant, mice, snake, peacock is there. Okay. And I am saying that basically 30 joules of energy is trapped in the producer level. How much is available for what? For the peacock, Baba. Can you guess? How much is available for the peacock? Okay, Sumit says it is D. Sagar says the answer for this question is B. It's Zainab says B. Queen says A. Bharti says A. Sorry, Bharti says B. Okay, Nisha says B. Rahul says okay. Q. No. Okay, so most of you guys are saying the answer for this question is B. And Vijay says 0.03, very precise. Okay, so the answer could be this. Do you think so? Let's make it out. So whenever you have a question like this, make something like this and then give your answers. So what do you have? You have a plant, you have a mice, and you have a snake, and you have a peacock. Okay, you have to find what is there for the peacock, right? And here you are saying 30 joules of energy is there over here. 10% law has been applied, only 10% things are going to go up. So let's go. 10% of this is going to be what? 0 cancel, you just have got. So only 3 joules is there over here for this particular trophic level. Now 10% of this would be what? 0.3 joules. 10% of this would be what? 0.03 joules. And that is the question being asked, that at the peacock, what is available to the peacock? Basically 0.03 joules is the only thing that is available for the peacock, which is why the answer for this question is going to be B, macha. Okay, it is going to be B. So I know that it sounds silly to make a diagram like this, but it could be helpful for you. Okay. Ma'am, can we mark the answers? I have to check. Okay, I have to check. Ma'am, I can't understand your handwriting. Quite usual. No one can understand my handwriting. I'm still working on it. You know what? I'll tell you something, okay? So when I write in paper, this is an extra knowledge, a little, what do you call, out of the bus. When I write in paper, I have a very beautiful handwriting, okay? When I write in paper, not over here, okay? But then no one understands what I'm writing. So once, when I was doing UG, okay, in my UG, I got 100 in one particular subject, but there was a side comment written over there, and that is handwriting very beautiful but not readable. So I think because he was not able to read, he gave me 100 on 100. I do not know what reason, but yes. So yes, guys, I hope this question is clear, and let's go with the next question out there, and that is question number 19. By the way, this question is from the chapter ecosystem. Okay, question number 19. Dash the procedure through which a piece of DNA is introduced in a host bacterium. Dash is a procedure through which a piece of DNA is introduced into a host bacterium. What is the procedure? Transcription, translation, transformation, transportation. Shreya says the answer for this question is C. Noah Kazi says C. Deadpool says A. It's Zainab says C. Okay, Sumit says Z. Bharati, Rohit Gowda, Vijay. Sorry, Divya Prajapati. I've changed the name also. Okay, and Darshan, GS Suni, Sri Ganesh. So many children are there, and the answer for this question, as you say, it is what? Transformation. So dash is the procedure through which a piece of DNA is introduced in the host bacterium. That process known as transformation. I just told you right now. So a cell is not having any plasmid. Now if a cell is having a plasmid out there, basically the process of taking it in is known as transformation. I'm getting transformed into something new. So the answer is pretty simple. Transcription, translation has nothing to do over here. Transportation is something else. So basically the answer is simple. So which is why I'm going ahead with the next question. By the way, this question was from the chapter Biotechnology, Principle and Processes. Okay, the gene producing dash was introduced into the first transgenic cow. The gene producing dash was introduced into the first transgenic cow. What do you think is the answer for this? What do you think is the answer for this? Ma'am, when we get translation is usually for the DNA to RNA, RNA to protein. That is what you call as translation. No, macha. And everyone who is asking me for the dress code, the reason, listen, I know that several YouTube channels are making up the video. I'm not making the video because for the reason that they usually publish the dress code. I'm just waiting for an official statement. That will be better, right? For the safer zone. But still, I'll check. Yes. So most of you guys are saying the answer for this question is A, and some of you are confused with B also, which is why I have to make my entry. Let me make my entry. So a transgenic cow is there. If at all you remember the name of the cow's name is I am Rosie. Rosie was the name of the cow. Now, what gene was being introduced? Human alpha-lactalbumin. Why? Because this alpha-lactalbumin is a very important protein, and basically it makes the milk rich in something that is important first. So this is produced by human beings in general. This protein is made in the case of human beings. But now when you pass the gene to a cow, the cow will be producing this in their milk. So obviously that is really, really good. And if at all a cow is able to do that, it will be a large-scale production of it, right? So the answer for this question is this. Why not alpha-1 antitrypsin? Because alpha-1 antitrypsin is not for that reason, not for increasing the nutritional quality. What is the use of alpha-1 antitrypsin? Any idea? What is the use of alpha-1 antitrypsin? Any idea? Cow name was also Rosie. Yes. Whose name is Rosie? Emphysema, exactly. Alpha-1 antitrypsin is used to treat a respiratory disorder which is known as emphysema. It is known as emphysema. So over here it is different. It is just human alpha-lactalbumin. If it is clear, just give me a thumbs up. If this particular question is clear, just give me a thumbs up. My dear, I have not had my food. Okay. Someone was asking me, ma'am, tiffin? No dear, I have not had. Okay. Now question number 21. Basically this is from PO1, and by the way, this was from the chapter biotechnology applications, biotechnology and its applications. Okay, come on. Arrange the following in ascending order of linane hierarchy. Ascending order of linane hierarchy. What do you think? Ascending means lower to higher. D, A, D. So most of you guys said A is the answer, which is why I showed. What is the question? I also told once more. Ascending order. Ascending means what? Low to high. Little bit of mathematics in the case of biology also. Low to high. Low to high means you do not have to look from kingdom to species. Rather, you have to look from species to kingdom. So you know the answer, but you're getting it wrong. Why? Because you just didn't read this. So make sure you read this. And for those of you who do not remember at all, I have a way for you guys to remember. Listen to me. King Peter came over for a great soup. King Peter came over for a great soup. So if at all you have to take it in ascending order, first comes species, then comes genus, then comes family, then comes order, then comes class, then comes phylum, and finally comes kingdom. So ascending and descending is something important. Do not make mistakes out there. And if this particular question is clear, just yes, keep the pot clean or family gets sick. That is also a good trick, Shreyas. Yes. I have heard about it. That is also a good thing. Pranit and okay, Tarun, everyone came up with their mnemonics. Perfect. So I think we can go ahead to the next question. Shall we? Samprit, we'll come down. Don't worry much. We are on 21st question. We have more questions, right? You will see 60 questions. This is a mock test. 60 questions will be there. You will be able to find PU1 also. Okay, let's go ahead. The development of young embryos of pteridophyte. Okay, so this was from the chapter, you know that. Okay, it was from the living world. And development of young embryos of pteridophytes within the female gametophyte is the precursor to what? Precursor to what? Aquatic habitat, at autotrophic habitat, and seed habit or parasitic habit. What do you think is the answer for this particular question? Darshan says 22B is the answer. Vijay says it is C. Guys, please make sure you read the question carefully. Shankar says it is D. Vijay says it is C. Okay, Fatima says C. Kavya says A. It's Zainab says B. Sandy says A. Sampreet says B. Guys, look at the question carefully. What are pteridophytes? What are pteridophytes? Listen, you had bryophytes, okay, which did not have the xylem and the phloem. They did not even have a proper plant-like structure out there. Slowly you got pteridophytes. They were the first one to have an advanced vascular tissues out there, right? And from them came gymnosperms. Gymnosperms were the one who actually had seeds. So just before them, if at all you're talking about pteridophytes, and if we are saying that the young embryos which are being formed, they are basically inside the female gametophyte, that means basically they are preparing themselves for what? For a seed habitat, for a seed habit. Okay, that's when they gave rise to, they later on diversified to form what? The gymnosperms out there. So the answer for this question is going to be C. It is going to be C. Precursor as in the starting point, the previous one, the precursor to the next life form out there. The first point through which you get the, for example, I say there is a cell and this is a precursor to the next cell. That means this cell is the one that is further going to give me rise to the other cells out there. That is what you mean by precursor. Okay. Cool. Yes, I am answering seed habit from that time. Yes, Risha, you're perfect. Correct. Oh my God. Okay, so yes. So guys, everyone who is giving me the answer, Prasad, it's still not been sorted. I had actually escalated this and yeah, I'll still look into it, okay? So the answer for this particular question is C. I hope all of you, it's fine. Guys, if it's clear, can you quickly give me a thumbs up? Can you quickly give me a thumbs up? And just give me a second. Just give me a second, okay guys? Very important thing. Okay. Next question. Question number 23. Release toxins of which group of protista is the cause of the death of other marine animals, such as fishes? Any idea? Any idea? Release toxins from which group of protista? So there is someone in kingdom protista who basically release certain things that kills the entire marine organisms out there. Any idea who is that? BB, Vandana, Rishika, it's me, Zainab, Shankar, Sumit, Jadhav, RJ Patil, and Sharanya, Noho, Kazi, Darshan, Lekhana, and Vandana, Dikshit, Deadpool, Bharti, and where is, okay. Perfect. So all of you are saying the answer for this particular question is going to be B. B is what? Dinoflagellates. It is dinoflagellates. You are correct. So there is a thing given about dinoflagellates that you should be knowing, and there is a what do you say? They have clearly mentioned it. Very often, red dinoflagellates, example, Goniolix, okay? Goniolix undergo such rapid multiplication that they make the sea appear red in color, okay? The toxin released by such large number may even kill the other marine organisms out there. So it may look nice, but actually it's very, very deadly, okay? So please remember the answer for this particular question is going to be dinoflagellates. This is a very unique thing about them, and yeah, that's all. Cool. Anything else that you need to remember? They have two flagellas usually, okay? They have two flagella. One lies longitudinally and other which is there, it is inside the wall plates that is there. So they have wall plates and stuff like that, which makes them very unique. Cool. So I hope that works. And now, by the way, this is from the chapter Biological Classification, and now we go to the next question, question number 24. Bulliform cells are the characteristic feature of what? Bulliform cells are the characteristic features of what? Someone was Trace is asking what's the pigment causing that color. It can be multiple. There are multiple pigments. One that I remember is pyridine. Okay. So Bhavish says 23B is the answer for this question. Okay, where are we? We are doing some other question. Okay, so most of you guys are saying the answer for this question is C. Okay, it is C. C is what? C is grass cell. Do you think that bulliform cells are the characteristic feature of grass cells? This question is important. Why? Because there was a PYQ on the basis of this also. That is why this particular concept is important. If at all it's not clear, make sure it is clear to you right now. What are bulliform cells? So usually in the case of monocots, okay, usually in the case of monocots, and you know grass is a monocot, in the upper surface, okay, adaxial surface is what do you call it? Adaxial surface is what do you call it? You will be having certain epidermal cells which are specialized, okay, they have become thin walled in nature, and these cells are known as bulliform cells. They are found mostly towards the adaxial surface, that is the upper surface, and they are thin walled epidermal cells out there. What exactly happens? What is the use of them? They are responsible for making sure that the water content is maintained out there. If at all, suppose there is too less water out there, so what is going to happen? These bulliform cells which are there, they are going to lose the water, and when they lose the water, they will make the leaf curl up. They will make the leaf curl up, and when the leaf have curled up, do you think that sun can directly come in contact with the stomata and stuff like that and make it lose more water? No. That is why curling is very important. So in that particular case, bulliform cells will be losing the water. Now, on the other hand, my dear children, when enough water is there, they remain turgid, and that makes sure that the leaf is also completely open, such that the process of photosynthesis and everything can easily take place. So that is the role of bulliform cells. Please remember, they are usually found in the case of what? Monocots. Leaves and the monocots are there. Large, empty, colorless cells. Exactly, Nandini. Okay, exactly. Perfect. So they help in leaf folding and unfolding to reduce the water loss during the dry conditions. When water is abundant, bulliform cells are turgid. That means they have enough. They are like this, keeping the leaf open for photosynthesis, whereas during water scarcity, they lose the turgid pressure, reducing the transpiration. I hope it's okay. Right? I hope that works. Guys, if at all it's okay, just give me a thumbs up. Everyone. And by the way, I could not read out any of your names, so sorry. I'll just read it out first. That's my thing to do. So Sharmila, Chandana, Rajita, Nisha, RJ Patil, Atif, Prashant, Pranit, Anand, RJ, and Bharati, Sri Ganesh, and Manoj Gowda, Shankar Nayak, Deadpool, Pallu, Nisha, Tarun, Sri Ganesh, Kalpana, Gayatri, and Tarun, Nakshatra, Nikita, Rohit, Shankar, and I think most of them are done. Yes, Naveen, everyone, Hasi, I think, yeah, Hasina. So, and M3Y also, everyone. I hope this is perfectly fine. Yes, cool. Now let's go on with the next question that is out there, question number 25. And the flower shown in the above picture is, the flower shown in the above picture would be what, Baba? It is going to be hypogynous flower, epigynous flower, perigynous flower, or the China rose. So this is the picture that is given to you. Look at this picture very carefully and then only give me the answer. Soumya, ma'am, Soumya. Okay, Shraddha says Soumya. Okay, Soumya is a name. Hi, Soumya. Okay. So, ma'am's famous dialogue. Cool. Yes. Yes, Macha. So, all of you are giving me the answers A, except some people who are saying B is the answer for this particular question. And some people are saying C. So, most of you guys are saying it is A. Why A? Why A? Dun, dun, dun. And my children are giving me wrong answers. Why, Ramacha? Why? The flower shown in the diagram, if at all you look at this particular diagram, what can you clearly see? This is your ovary, right? Ovary is down, and over here from the top level you have what? The petals and the sepals and everything coming up. Ovary is down. What do you call this condition? Epigynous condition. No. What do you call this? Epigynous. You denote it by this particular thing. This is epigynous condition. On the other hand, if at all you had the ovary right over here, okay, this is the ovary, suppose, and this is the stigma and this is the style, okay, and this is the thalamus. From here, if at all you're getting petal, the sepal and everything, you will call this hypogynous. The ovary is dominant out there, okay, it is superior out there. That is what you mean by this particular symbol. Over here, it's the ulta, so it is going to be epigynous. B is going to be the answer, na macha. Why are you saying A is the answer? And what do you mean by perigynous? Where it is all of the on the same level. For example, your ovary is over here, stigma style is right over here, in the same level, somewhere over here. Okay, somewhere over here you have the petal and the sepal. So this is somewhere in the middle, which is why you call this as perigynous condition. This is epigynous, it is down. Okay, this is hypogynous, and this is going to be perigynous condition. Machi, yes, Sampreet. So the answer for this question is going to be what? B. It is going to be B. Very, 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 very important. Okay, clear na? Clear, everyone. Will you forget it now? Will you forget it? You should not, okay? Someone is saying, I love you, Nakshatra. What is happening over here? Okay? Okay. So, yes. If it's clear, just give me a thumbs up. Super clear. Okay, thank you so much. No, ma'am. For those of you it's not clear, I'll tell you once more. Please listen to me carefully. This is an important concept. Questions have come from here. Okay. As Harish is so papa guy says, I love you, Nakshatra, but only as my sister. Good boy, good boy. Okay. So listen to me carefully. You have over here ovary, stigma, and the style and the stigma, right? The condition where you have the thalamus, and basically you have the petals from here, and you have the sepals from here, and basically you have the male part from here. What do you understand from this? At the top of all of this thing, you would be having your ovary. So I'll say the ovary is what? Superior. I'll say it is superior, so I'll call this condition as hypogynous condition. Hypogynous condition, it is denoted by this particular thing. Okay, it is not by on the top of dash you have G. That means on the top of dash you have the gynoecium. That is what you mean. Now the next condition that is there is basically this, where you have your ovary, style and the stigma, right? And it's all being covered. From here you have your petal. Just a second. From here you have your petal, your sepal and everything out there. So basically we can also the male part. In this particular condition we say that it is down. The gynoecium is basically towards the down. You call this condition as what? Epigynous condition. Epigynous condition. This is basically the inferior type of ovary. Inferior ovary. Now one more thing is there, perigynous. Perigynous means all of them are going to be in the same level. The ovary will be there. In the same level you will be having the petals being joined and also the male part being joined. When it is on the same level, you say that it is perigynous in the middle way. Okay, cool? Yes. Now going ahead with the next question, and that is, photosystems are functional units present in what? So by the way, this question is from the chapter Morphology of Flowering Plants, and let's go ahead with question number 26. And what is happening over here? Guys, if at all you are not studying over here, I have the ninja technique of putting you in timeout and also blocking you. So please do not do that. Okay? Yes. So everyone who is over here, make sure you solve the questions. Just the questions, and we can have a chit chat between amongst each other. Yes. Ma'am, please continue. Most repeated PYQ. PSR, dear, I have already completed the PU2, all the PYQs out there, right? So, yeah. And we will be solving more towards the complete revision. So, you are going to start, by the way, everyone, I hope you know that we are going to start the complete revision series. Yeah, from today onwards, you have the first series that is for mathematics. Complete revision will be there. If at all you are a maths candidate, go check it out. Okay. So, most of you guys are saying the answer for this particular question is B. Some of you are saying it is C. Photosystem of functional units present in. You have photosystem, two photosystem, PS1 and PS2, right? And they are not present in the stroma, Baba. They are going to be present in what? The thylakoid membrane. No, C is the answer for this question. Thylakoid membrane. How can it be present in the stroma? Listen, this is your chloroplast. You have the thylakoid over here stacked up together to form the grana. and the space that you have over here is known as the stroma. On the membranes of this thylakoid, you would be having what? Both the photosystems out there. Always remember, it's not the inner membrane of chloroplast or stroma or the outer membrane of chloroplast. The only answer for this question is going to be thylakoid membrane. So the answer is C. So those of you who are saying A and all, macha, we are getting things wrong, okay? The answer is going to be C. Cool. Cool. If it's clear, just give me a thumbs up. If it's clear, just give me a thumbs up. PS1, PS2 in the thylakoid membrane. That is why light reactions are going to happen in the thylakoid membrane. The dark reactions happens in the stroma. Okay. Trisha, ma'am, I have already forgotten photosynthesis and respiration of plants. What do? I'll recommend you, if at all you think that that is the only chapter which is like, you can spend some time to just go through a quick one-shot video of that particular chapter and solve the PYQs. That will be really, really good. Okay? So do one shot. Okay, let's go. Now the next question that we have is question number 27. Select the correct order of reactions in glycolysis. You have to find the correct order of reactions in glycolysis. So again, when you get these type of questions, what do I say? Read the options carefully, and the option that you're pretty sure that will be the first one, mark it to be the first one. So conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate comes a bit late. Conversion of 3-phosphoglyceric acid to 2-phosphoglycerate. Conversion of BPG, that is bisphosphoglycerate, to 3-phosphoglycerate comes somewhere in the middle. Splitting of fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This is the first guy that is going to be there. So this is definitely going to be the first one. Wherever I see options where it is going to be starting with D, I am going to choose that particular option. Right, Baba? So this option and this option is going to be eliminated. This is the option that I will look forward for. Now I can choose either of them and I can begin from. Okay, suppose I am choosing this one. After D, A is mentioned. So let me see. Fructose 1,6-bisphosphate is mentioned. Fructose 1,6-bisphosphate is broken down, or basically is being converted to what? Dihydroxyacetone phosphate and also glyceraldehyde 3-phosphate. This is the thing. Okay, this was the first step that has been taken place over here. Now what do you do? According to this, you go with A. Conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Yes, so this is right over here, Baba. I can convert the glyceraldehyde 3-phosphate to the next thing, that is what? 1,3-bisphosphate. Can I do that? Yes. But according to this, what is the next step? Conversion of BPGA. Bisphosphoglyceraldehyde. We have not got bisphosphoglyceraldehyde, so how can we convert it? So obviously that will not be the answer. Your answer is going to be D itself. Your answer for this question is going to be D, and I know D, Machi, all of you are saying D, 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 D. Some of you are saying A is the answer. Why? D is the answer for this question. No. Okay, so now if at all we do not waste our time and we look at this particular diagram, we'll get more idea of it. For those of you who have forgotten glycolysis, listen to me carefully. First thing that happens is glucose molecule is there. Glucose molecule contains how many carbon, guys? How many carbons are there in one glucose molecule? Come on, everyone. How many carbons are there in one glucose molecule? 6, exactly, Tarun. Okay, so I'm explaining right now. Listen to me very carefully. 6 carbon is there in one glucose molecule. Fine. Now over here, suppose I am adding one phosphate group, so it becomes what? Glucose 6-phosphate. On any of these carbons, I am adding one phosphate group. It becomes glucose 6-phosphate. Now there should be someone to give me a phosphate. Who is that someone? Obviously ATP is going to be giving me the phosphate. When ATP gives me the phosphate, it transforms into ADP. So adenosine triphosphate, as in it has 3 phosphate. But now it is being adenosine diphosphate. It has lost one phosphate to what? To glucose. It has given one phosphate to the glucose. Okay. Now glucose 6-phosphate, it isomerizes to what? Fructose 6-phosphate. Glucose 6-phosphate isomerizes to fructose 6-phosphate. Just little change out there, right? And then the fructose 6-phosphate says I need one more phosphate. Macha, give me one more phosphate. So one more phosphate is also getting added up. Again, where can it get phosphate from? You have to borrow it from ATP only. One ATP gives one phosphate. Now it is not fructose 6-phosphate, it is fructose 1,6-bisphosphate. What do you mean by that? 1 at the first position and at the 6th position it has a phosphate group, and since it has 2 phosphate, I will call this bisphosphate. Okay, fructose is 1 bisphosphate. Until this, is it clear to all of you? I hope it is clear, right? Okay, so we have got this. Now listen to me carefully. Fructose 1,6-bisphosphate, which is there, It's again a 6-carbon compound. This 6-carbon compound can break down into two things. One with 3 carbon, one with 3 carbon. 3 carbon. So 3 plus 3, 6, right? So one that it breaks into is glyceraldehyde 3-phosphate, and the other one that it breaks is dihydroxyacetone phosphate. Since it has 2 phosphate group, it gave the 1 phosphate group over here, 1 phosphate group over here. So one is glyceraldehyde 3-phosphate, other one is dihydroxyacetone phosphate. Okay, it has got that also. Okay, everything is fine. But listen, for the further process of glycolysis to happen, you only need this guy. So what happens to this matcha? This matcha is of no use, no bubbles of use. It is basically getting isomerized back into glyceraldehyde 3-phosphate. So in easier terms, we can just say that there are 2 molecules of glyceraldehyde 3-phosphate being formed over here. There are 2 molecules that is being formed over here. Okay, now glyceraldehyde 3-phosphate, which is there, 2 molecules, please understand, they are going to convert into the next thing, that is 1,3-bisphosphoglycerate. 1,3-bisphospho, again you have taken a phosphate group and becomes what? 1,3-bisphosphoglycerate. Cool. 2 molecules of that also. Cool. Now again what do? You basically are going to lose some phosphate group. So it was 1,3-bisphosphoglycerate. This time you have lost the phosphate group. It has now become 3-phosphoglycerate. So you might be asking, if only 1 phosphate group is going, why do we need 2 ATP? Are Baba, you are talking about 2 molecules. No. So one guy lost 1, another guy also lost 1. Okay. So when you lose the phosphate, where does it go to? The ADP which was there says that give me back the phosphate. No, I will become ATP. So you got back your ATP also. So finally you got the 3-phosphoglycerate. Again 2 molecules. This guy converts to what? 2-phosphoglycerate. The position is moving. The phosphate group has moved from 3rd position to the 2nd position. Now the 2-phosphoglycerate converts to what, Baba? Phosphoenol pyruvate. Phosphoenol pyruvate. Okay, there are again 2 molecules, and finally it gives rise to the pyruvate. Here again it will be losing the phosphate group. So finally you are getting pyruvate, 2 molecules of them. Okay, you get 2 molecules of them. So I know it's not a one-step process that you can remember very easily, but I'll recommend you to please remember the glycolysis are there, the steps of glycolysis. It's important, very, very important. Yeah? Clear now? Ma'am, teaching is so nice. Thank you, Amrish. So your answer for this question is DACB. As I said, please look out for the option which will be anyway the first one. So I spot that the D1 was the first one. So I looked for the D option which is first. So this and this came. Now from the options itself, basically I'm going to check my answer over here. Cool. So if it is clear, just give me a quick thumbs up. So those of you are asking break, I'll give you a break. Okay, I'll give you a break when we reach 30th question. Once we're done with 30th question, I'll take a break. Okay. Thumbs up, guys. Thumbs up, thumbs up. Yes, perfect. Let's go. Which is not a step in the Calvin cycle? Which one is not the step in the Calvin cycle? Carboxylation, reduction, regeneration, photophosphorylation. Carboxylation, reduction, regeneration, photophosphorylation. Now anyone who is there, who is stuck in some big questions, I'll request you guys, if at all you could stay back a bit. Once after the class ends, you can ask me the doubts. I will only go after solving the doubts. Okay, that's my promise. I've always done that. Yeah? And yes. Hiya. RJ Patil, Deadpool, Sampreet, Sumit, Hema, Akshita, Girija, RJ Tarun, Ismi, Zainab, everyone. Sandy, Malikarjun, Chandana, Rohit, Gayatri, Ramya, Malik and Pavitra, all of you says, and M3Y says, the answer for this question is Sia. Sia, guys, check out the answer. Check out the answer very clearly. Which is not the step in Calvin cycle. Calvin cycle means dark reaction. Dark reaction, right? The answer for this question is going to be D, photophosphorylation. How can it be regeneration? Regeneration is happening if at all you look at the process. If at all you look at the process, my dear children, the first process is carboxylation. Where what happens? Basically CO2 which is there, okay, it has been taken up. It has been fixed using an enzyme that is Rubisco. Yeah. So you had RUBP. RUBP plus CO2 comes together to finally form what? 3-PGA, which is known as 3-phosphoglycerate. Okay, 3-PGA is being formed. So first process is carboxylation, where you fix the carbon out there to produce this particular thing. Then you see reduction. Why? When do you see reduction? When 3-PGA, okay, is converted to G3P, that is glyceraldehyde 3-phosphate. Over here, Baba, you are going to use you are going to use NADPH, right? You are going to use NADPH and you will clearly see reduction out there. So this is also not the one. Okay, now you have got G3P, that is glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate, yes, it is utilized for the production of glucose, but some of it is used for regenerating what? Regenerating RUBP. Regenerating RUBP. So ribulose bisphosphate. So please remember that also thing is also there. So regeneration is also happening over here. But photophosphorylation is not there. What do you mean by photophosphorylation? It means formation of ATP using light energy. Photo means light. And phosphorylation means formation of the ATP molecules out there, right? So light energy basically does not have anything to do over here. It is a dark reaction, and obviously here you're not generating ATP, rather you are going to utilize the ATP. You're not generating, you're utilizing the ATP, which is why the answer for this question is going to be D. Okay, is this clear? If it's clear, just give me a thumbs up. If it's clear, just give me a thumbs up. Photophosphorylation happens in the light reaction. Cool, na? Yeah. Okay. Next question, question number 29. The hormone that promotes rapid elongation of the internode or petiole in deep water rice plants is? In deep water rice plant. Any idea? The hormone that promotes rapid elongation of the internode or petiole. Internode as in, for example, if at all you look at this particular thing, okay, suppose this is a plant. Over here I have a node from where I got a branch, okay. Here there's another node from where I got a branch. This particular area between two nodes is known as internode. And now you are saying that there is a hormone that is going to promote rapid elongation, okay. So node is over here, another node is over here. You are elongating this particular area. Which is the hormone which is responsible for the rapid elongation in deep water rice plants? D. Ma'am, it's D. Why are you not looking at the answer is what you might be assuming. So D, gibberellina. Who was that person? Tarun says D. Okay. Malika says D. Sandy says C. Guys, I think you are confused. I'm not talking about roseate plants. Okay, you might have seen this rapid elongation thing in the case of roseate plants, where gibberellin has a role to play. But over here, things are quite different. Please check out your answers. The answer for this question is going to be B. And some of you gave me the answer B. Vishwant, I can clearly see B. Vandana gave me B. Panvit gave me B. Mallikarjun and Deadpool gave me B, and so there are some children who gave me the answer as B. Chandana also. Guys, if at all I'm missing out your name, I'm so sorry. Guru Music, everyone. Vaishnavi. So the answer for this question is going to be B. I'm not talking about gibberellin. Gibberellin is in the case of roseate plants. You might have seen that they are responsible for something else related to this only. Okay, but this is ethylene. And what exactly happens in the case of ethylene is that ethylene, you know, it's a gaseous hormone, right? It's a gaseous hormone out there. And basically, when the plant is in the submerged condition and the gas is being released in the case of in the water, what exactly happens? The gas will not be able to move around. It is trapped. It accumulates. When it accumulates, what is going to happen? Basically, it is going to lead to the elongation of those areas where it is being accumulated, especially the intertonal regions. What is the use of it? Basically, suppose over here I have the rice plant. Okay, this is a rice plant. If at all the water is filling up until this particular level, it's totally submerged. In that particular case, obviously the plant cannot operate. So it has to make sure that the plant outgrows this. The plant outgrows this. That is a major role that is done. So basically, that is the role of ethylene. Ethylene is a gaseous hormone. It accumulates in the water, and basically it leads to the internodal region elongation, rapid elongation. Cool, na? So everyone who gave me the answer D, I hope it's sorted now. Is it sorted? Chandana ma'am, you're so sweet. Thanks, bacha. Sorted? Clear? Will you forget it now? Can you tell me what does gibberellin do? Anyone who remembers about gibberellin? What does it do? Any idea? So gibberellin in the case of roseate plants, Baba, it leads to bolting. Bolting means it does not allow the elongation of things. It makes sure that it is only small. Okay, so that is known as bolting. So yeah, and that is why the leaves are being clustered like this in the case of cauliflower and things like that. Bolting, exactly. Okay, so gibberellin is for bolting. This is something else that is being discussed over here. Cool. Question number 30, and after this question number 30 we are going to take a quick break. And by the way, everyone who have not clicked on the subscribe button and not hit on the like button, do it right now. Okay, are you not liking the class? Yeah, so yes, when tripalmitin is used as respiratory substance, the value of RQ is what? Respiratory quotient is what? When tripalmitin is used as a respiratory substance, these are all the most repeated type of questions coming from the PU1. Make sure you go through them. The answer for this question is what? Shreya says the answer is D. Okay. Vidya says it is D. Zainab says it is image growth. Okay, I think the previous question you were answering. Mallikarjun says D. Hasina says D. Panvit, all in one. And Naveen, GS Suni, almost everyone says the answer. Deadpool says the answer is D. Tick, 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 tick. Sumaya, Deepan, Trisha, okay, Vidya, okay, Vaishnavi, everyone out there. If at all I'm missing your name, I'm so sorry. Nakshatra, Ramya, everyone, Mellow Universe. Okay, so the answer for this question is going to be D, and you have to remember it. Okay, you have to remember it. First of all, what do you mean by respiratory quotient? It is having a, what do you call, a formula. Respiratory coefficient is equals to amount of CO2 evolved divided by the oxygen consumed. Okay, amount of CO2 evolved and the basically oxygen which is being consumed. So in the case of tripalmitin, tripalmitin is what? Is a fatty acid. It's a fatty acid. So you're talking about fats. Listen to me. Fats, carbohydrates and proteins, all of them have different respiratory coefficient, sort of fixed respiratory coefficient which you have to remember. So in the case of fat, it is usually 0.7. It's 0.7. Why? The reason being that tripalmitin is a type of fat, undergoes fatty acid oxidation, which requires more oxygen than the amount of carbon dioxide produced. So over here, the amount of oxygen which is consumed is more, rather the amount of CO2 which is evolved, which is why you get a decimal answer that is 0.7. That is 0.7. Always remember the formula. Formula is important, and also this particular thing. Now, in the case of carbohydrates, what is the answer? In the case of carbohydrates, Baba, it is said that the RQ will be 1. In the case of carbohydrates, the CO2 evolved and the oxygen utilized, all of them comes out to be equal. So it is going to be 1. Okay, what about the proteins? When you're talking about proteins out there, proteins, the ratio that you will be seeing is would be about what? 0.9. 0.9. So if at all I am asking you this question once more, what is respiratory coefficient? Formula. Give me the formula for respiratory coefficient, everyone. What is the formula for respiratory coefficient? Everyone out there. Baba means child. Yes, exactly. I come up with a lot of things. Baba, Macha, this. I'm asking you, what is the formula for respiratory coefficient? CO2, O2. CO2, O2. Okay. CO2, O2. What is it? CO2, O2. CO2 evolved or consumed? CO2 evolved or consumed? What do you think? It is going to be the amount of CO2 which is going to be evolved and the oxygen which is going to be consumed. In respiration, obviously oxygen is always consumed and CO2 is something that is being evolved. So you do not have to do rectification. Understand it. Okay, oxygen is being consumed and CO2 is being evolved. Okay. Now if at all I am talking about carbs, what is the respiratory coefficient usually? Sorry, not coefficient, quotient. Respiratory quotient of carbs is what, Baba? Shankar, Nayak, Arif, Sumit, as well as Guru, Gagan, and Deadpool, everyone. It's me, Zainab, Deepa, Tarun, Anant, RJ Patil, Darshan, Ravindra, Nakshatra, Chetu, everyone perfect. Come on. 1. It is going to be 1. Carbs is equals to 1. Okay, what about fat? For fat, it is going to be 0. What? For fat, it is going to be perfect, Hitesh. It is going to be okay. Rakshita says for fat, Mallikarjun, everyone, Vandana, Rakshita, everyone says it is going to be 0.7. It is going to be 0.7. For those of you, if at all you have said 0.9, addigittum. Okay, it is going to be 0.7 for fat. Okay, now what about proteins, Baba? For proteins, it is going to be 0.9. It is going to be 0.9. I'll tell you a way to remember it. Okay, will you remember it? Look here, it is P, and P looks like ulta 9. P looks like ulta 9. So 0.9 for proteins. And fat, in the fat, Baba, it looks like 7 only, no? So 0.7 over here. I discovered it right now. Okay, so yes. Clear na? Did you like this? Okay. Perfect. Wow, what a trick. Yes, thank you, thank you, thank you. Yes, so guys, I think we can take a break since everyone is tired, and so let's take a break of 5 minutes and come back. 11:13 is the time. We will come back. Let's take a Kit Kat break and be right back. Bye. Okay, 30-minute break. Only 5 minutes. Come on, guys. If at all I do not end it up, you will only scold me, right? So yes, better. So I'll also go take some rest and come back. So, taking break. Hello, 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 hello. Is the break done? Because I was seeing, oh my god, give still 5 more minutes. Listen, I can give you 5 more minutes, I don't mind. But the thing is, you are the ones who will tell me that it's late. Okay. Some people were talking about Dhurandhar. Have you watched Dhurandhar Macha and all? Oh my god. Okay, chalo, let's go ahead then. Let's go ahead, no? I know it could be a bit hard, but do we have much time, actually speaking? No, right? So we can go ahead. Yeah, then we can take a break. 30 more questions, Baba, and then we are finishing up the first FLT out there, full length test out there. So let's go ahead, and the first question goes like this after the break. 31st question, let's see. Select the correct statement. India has more than 50,000 genetically different strains of rice. India has 1,000 varieties of mango. At ecosystem level, India with its deserts, rainforests, mangroves, etc. has a greater diversity than a Scandinavian country like Norway. The tropical rainforest initially covered 14% of the land surface, but now covers only 6% of the land area. What do you think is the answer for this particular question? Hello Trisha, hello, hello everyone out there. Okay, come on. Trisha says the answer for this question is D. What about others? Tarun says D. Pallavi, yes dear, Macha, tell me ra. Okay, Nauman says it is D. Vishwan says it is B. Why B? Okay. MS Bindushri is also there. Pallavi, Deadpool, Rohit, Sumit, Melo, and Darshan, and Anant, and Tarun, Arif, Ramya, everyone says. Most of you guys are saying it is D, but some people are saying it is B also. I think there's a slight confusion out there. The answer for this question is D. Why D? Listen to me. So this is an example of genetic diversity that is given in your NCERT. It says that they can, you know, in mango, you go to Kerala, you will have different type of mango. If you go to the north, you will have different type of mango. Different strains of mango are there, right? So it's mango only. It's Magnifera only, but the thing is that species, if we talk about genetic level, it is kind of different, right? So 50,000 genetically different strains of rice, is it there? Yes, it is actually there. 50,000 genetically different types of rice is there. This is coming under genetic diversity. Genetic diversity. This and mango, everything comes under genetic diversity. Now, India has 1,000 varieties of mango. Again, genetic diversity. It has 1,000 varieties of mango. Now, next one. At ecosystem level, India with its desert, rainforest, mangroves has greater diversity than Scandinavian country like Norway. Yes, India has all these things, which is why we say that there is ecological diversity. This is an example of what? Ecological diversity out there. So there are 3 types of diversity: genetic, species, and finally ecological diversity. Species diversity has not been mentioned over here. Now the last statement is the tropical rainforest initially covered 14% of the land, but now Baba, only 6% of the land is being covered with the help of that. Why? Because a lot of things happened, right? Which is why the only answer for this question is going to be D, because D is the one which says 1, 2, 3, 4, everything is correct. Now the numbers over here are strictly very, very important. Make sure you remember that. And can anyone tell me what is the example for species diversity which is given in your NCERT? Example for species diversity. Tropical rainforest, someone is asking me to explain D. Tropical rainforest initially covered 14% of the land surface. Listen, so it was covering majority of it, 14%, but because of deforestation, because of lot of exploitation that we did, Baba, it is now covering only 6%. That's the explanation for this. Pallavi says, ma'am, today is my birthday. Happy birthday, Pallavi. You know what, today is my mother's birthday as well. So, yes. Divya, my name is Jasmine, dear. Okay. Guys, did you understand it? And I'm asking you a question. I am asking you a question. The question goes like this. Give me an example for species diversity. I spoke about genetic diversity. You have rice and you have mango and you have all that. I spoke about ecological diversity. That is India and Norway. India is more ecologically diverse with all this thing. Okay, someone said, Tarun says Western Ghats. Exactly. There is an example given in NCERT for species diversity of Western Ghats and Eastern Ghats. Okay, so basically, No, Rauwolfia vomitaria again comes under genetic diversity only. Rauwolfia comes under genetic diversity only. So Girish, Girija and also Uday, everyone is saying. What is there in Western Ghats? Amphibian species. So said that there are some amphibian species that are going to be more in Western Ghats in comparison to Eastern Ghats. That is the example for species diversity. Okay, so there are more species of amphibians in the Western than in Easterns. So always remember that. This is an example that you need to remember. One more is Rauwolfia vomitoria, that is for genetic diversity. Rauwolfia vomitoria, if at all you have seen this particular plant, my dear children, it is going to produce different concentration of toxin, and since it is producing different concentration of toxin, we say that genetically they are different varieties that is found. That is why they also come under what? They come under genetic diversity. Cool? Right now, will you forget this now? This is important. Please remember, you can get questions on the basis of that also. In biodiversity and conservation chapter, anything can be coming up. Yeah, a very good morning, student. And Pallavi, I like your classes. Is it? Thank you so much, guys. If it's clear, can everyone give me a quick thumbs up? Quick thumbs up, everyone. Ma'am, please teach in Marathi or Hindi. Mayur, I wish I knew all these languages, but I do not know it. I mean, I know Hindi, but not Marathi. And if at all I speak, listen, I know, I can talk in, what do you call, Malayalam. I can understand Tamil. But the thing is that if at all I start talking in different languages, do you think it can cater lots of children out there? No. So most of you might be knowing English. Some of you might have understanding of broken English, but at least you know the language, right? Take the benefit of it. Learn from it. It is going to be helpful for you. Yeah, ma'am, where are you from? I am from Kerala, but I was born and brought up in Indore, Madhya Pradesh. So I'm half Northy, half Southy. Okay, question number 32. Can I start with it? Guys, yes, perfect. Statement number A, Typhoid Mary was a health worker. Statement number B, Haemophilus influenza does not cause influenza. What do you think is the answer for this particular question? Come on. Deadpool says the answer is B. Mayur says the answer is A. Jeevan says it is D. Okay, Tarun says it is D. Guys, be very sure about what you're answering over here. Saptami says it is B. May 3Y says it is B. Bhavish says B. Sorry, D. Sumit says C. Harish D. Sorry, B. Nauman says D. Okay, so you guys have different answers, and let me. Trisha says it is B. Okay, Manu says it is B. So let me go on with the answer which is correct. First one. Typhoid Mary was a health worker. No, Baba, she was a cook. And if at all you remember, why was she known as Typhoid Mary? Because she was transmitting the disease typhoid to n number of people out there. So she used to go to washroom, but without washing her hands, she used to come back and cook food for the people. And that is exactly how typhoid is going to be passed. Typhoid is caused by Salmonella typhi, and it's passed through what? This food and the oral routes out there, right? So yes, so she was a cook, not a health worker. So this is why the statement is wrong. Second statement, Haemophilus influenza does not cause influenza. That is correct. No. Haemophilus influenza, does it cause influenza? No, it does not. It does not cause influenza. Influenza different from pneumonia. Haemophilus influenza is responsible for causing pneumonia, not influenza. The name is influenza, but is not influenza. Okay, so please remember it. So this particular statement is also correct. It does not is being said. It is correct. So the only statement number B is correct and A is not. So your answer is going to be B. It's going to be B. It causes pneumonia. Exactly, Ramya. Exactly. Guys, is it clear? Is the question too tough? No, right? It's easy. Okay, just a little bit of understanding required, that's all. If it's clear, thumbs up, thumbs up, thumbs up. Ma'am, can you understand Malayalam? Yes, I can talk Malayalam, I can understand. I talked in Malayalam at my home. Yeah, that's my mother tongue. Ma'am, I'm suffering from fever. You should be taking rest. Then okay, perfect. Let's go ahead with the next question. Question number 33. The type of immunity that is present at the time of birth in humans is? The type of immunity that is present at the time of birth. So when we are born with it. Is it specific? Is it artificial immunity? Is it acquired immunity? Or is it innate immunity? Okay, Vimla says the answer for this question is D. Hitesh also says it is D. Zainab says it is D. Okay, Tarun says D. Hema and Rohit and Amulya, Bhavish. Most of you guys. Sateri is also there. Hello, dear. Andy Suleiman. I'm trying to read out at least few names. Alima. Okay. So the answer for this question is going to be D. It is innate immunity. Innate means something that is present with us from birth. And please remember, it is non-specific type of immunity. For example, if at all, the easiest way to remember it is skin. We are all born with skin, right? Yes, we are all born with skin. And over here, is it specific out there? It says that virus, you can enter, bacteria, you cannot. No, is it not like that? It's not at all like that. It's non-specific. It's common to all. It does not allow anything to get inside. And that is why we say that it is non-specific. And why we say that it is present from birth? Because it is present from birth. That is why it comes under what? Innate immunity. Specific immunity is basically acquired immunity. Acquired immunity is something that we acquire during our lifetime when we get some infection, when we get some vaccination. We acquire this type of immunity, yeah, and it is very specific. When certain type of antigen comes, only certain type of antibodies are going to be released against it, yeah. And artificial immunity, basically when we are providing artificially some, for example, we are providing antibodies from outside and things like that, that comes under artificial immunity. So the answer for this question is D. If this question is clear to all of you, can you give me a thumbs up? Can you please give me a thumbs up? Mayur, thank you so much, dear. Okay. Ma'am, can you send the PYQ of the last syllabus? What? I didn't understand. Listen, by the way, everyone who is watching this video and who is, once again repeat. Yes, I can repeat it. Listen. It says that what is the type of immunity that is present from the birth. Guys, can I take 2 minutes of your time to explain about immunity if it's not too much? Guys, if at all, I can take 2 minutes of your time to explain everything about immunity that is important. Is it okay? Is it okay? Yes, okay. So just listen to me out carefully. This could be helping you well for your examination. Yeah? And I mean, I'm not just explaining this question, I'm just talking about everything in general. So we have two types of immunity. One is innate and the other one is acquired. Innate means it is going to be there from the birth. Okay, for example, our skin, for example, our tears, there are certain cells out there in our body. Polymorphonuclear leukocytes, which you call as neutrophils, or basically you have natural killer cells. Natural killer cells. You have certain other cells, macrophages. All of them, Baba, they are a part of our innate immunity. It is there from us, there with us from our birth. Okay, and also we have certain factors which is being released by our cells. You call these factors as cytokines. Cytokines, some secretions which are being released from our cells. The example for the secretion, very famous example is interferon. So on the basis of it, you might have read your NCERT, that is physical, physiological and cytological and basically cellular barriers which are there, cellular barriers, cytokine barriers, physical and physiological barriers. Make sure you go through it. I am going to talk more about the acquired immunity. Acquired is something that you are going to acquire throughout your lifetime. And listen to me very carefully. Suppose over here I have Pappu. For the first time, Pappu is getting infected by a pathogen. What is going to happen? Pappu is going to have fever. Pappu's response is going to be very, very bad. This response for the first time is known as primary response. And this is definitely going to be slow because our body sees the antigen for the very first time. It does not know how to react. But the secondary response which is there, the second time the same antigen comes, okay, the second time the same antigen comes, our body is going to react very nicely. Why? Because there is something that is happening over here during the primary response. There are certain things that are happening over here. What is this thing that is going to happen over here? Just give me a second. Come on, wipe off. Okay, so the first time our body sees an antigen, what is it going to do? It is going to activate two types of cells. One is the B cell and one is the T cell, basically the B lymphocyte and the T lymphocytes. The B cells, when they are activated, they are going to produce antibodies. They are going to produce what? antibodies, very specific to these antigens. These are what? These are antigens. On the other hand, they also produce some very important cells which is there. What do you call these important cells as? Memory cells. What does the memory cells do? Memory cells basically stores the information that when this antigen was produced, I basically produced these type of antibodies, because antibodies are different types. There is G, antibody A, antibody M, antibody E, antibody D, gamed. There are 5 types of antibodies. So which antibody did I release? That memory of it will be stored in the memory cells out there. Okay. Now what about the T cells? T cells, when they are going to act, they are going to be activated, they produce different type of cells. For example, there can be T helper cells, okay, which goes and tells the other cell that there's some problem over here and you have to do this. This acts like a mediator out there. Then there is T cytotoxic cells, which directly goes and kills the antigen out there. So there are different type of T cells, and apart from it, T cells also have what? Memory cells. They also produce memory cells. So the first time when Papu, okay, when Papu came across an antigen, what happened? Our body was able to produce antibodies. Our body was able to produce memory cells, but in very low amount. The second time the same antigen came into the picture, hey Macha, I have seen you somewhere. Now it knows what to do, so it produces more antibodies, which is why we say the secondary response or the anamnestic response which is there, it is going to be on the higher side. Okay. Now, what do you call this type of immunity? You call this type of immunity, first of all, we are talking about acquired immunity. Why acquired? Because this is something that we acquire during the lifetime. Okay, when we move on, that's when we antigens and everything are going to come across. This is known as humoral immunity. This is known as humoral immunity, and this is known as what? Cell-mediated immunity. Why humoral immunity? Because Baba, the antibodies which are produced over here, they are going to mix up with your blood, and blood is a fluid, and the other term for fluid is what? Humor. So we say that basically antibodies are there in the fluid of our body, and these antibodies provide the protection in those particular fluid wherever it goes. Okay, so that is why it is known as humoral immunity. This is known as cell-mediated immunity. Why? Because usually the T lymphocytes tell the other cells to do the job. They are the ones who instruct the other cells, or basically sometimes they directly go and kill these cells directly. So since cells are being over here in the midway, we say cell-mediated immunity is the one that is being followed by the T lymphocytes. These are the 2 things that you need to remember. Now, suppose a question is there coming from your NCERT. Suppose I have taken kidney from Zambia. Okay, I have taken kidney from Zambia. My body is going to reject that kidney. Why? Because that is foreign. So my body thinks it is foreign. I have to basically kill the cells. Now, if at all I am saying that graft rejection is happening over here, which out of these cells are going to play a major role over here? Always remember, during graft rejection, it is going to be the T cells which are going to play a major role over here. T cells are going to play a major role over here in the case of graft rejection. This can be a question. Okay, next question that can be there. The antibodies that ma'am you were talking about, you said that there is GAMED, gamed, right? What are these antibodies actually? These antibodies are nothing but proteins. They are proteins. You call these proteins, you have a name for it, immunoglobulins. Immunoglobulins, which is why you say IgG, IgA. What does this IG stand for? Immunoglobulins. These are what? They are all proteins, Baba. Until now, whatever I have told, is it clear to you? Whatever I have told you until now, is it clear? Now, the first one is G, and the first one is the one that is passed from the mother to the fetus. How? Through the placenta. Through the placenta, this is being passed. Now, when the baby is outside, through the milk of the mother, you pass IgA. You pass IgA. A for Amma, you can remember like that. Is this particular thing clear until now? This is very, very, very important. Okay. Now, the next thing that you have to remember when I say that I am giving antibodies. So, there are 2 types of immunity again. If you go forward, one is active and one is passive. Active means you are relying on your body to produce antibodies. For example, I got an infection. Basically, my body produces antibodies against it. I am totally fine. Or basically, I get a vaccination. Vaccination is what? I am getting weakened pathogens. Okay, weakened pathogens. So basically, in my body, I am getting antigens in a way. What is my body doing? My body is reacting to it, is producing antibodies. That is vaccination. I am making sure that my body is aware that something like this can actually come up in different form. So I am producing antibodies. Here, your body is actively participating in producing antibodies. That is known as active immunity. On the other hand, Baba, if at all you are saying that my body is not going to produce any antibody, you give me antibody. That happens in the case of babies. Babies do not have much antibodies out there, so the mother is providing antibodies. Yeah, naturally that is happening through the milk, but it can be done artificially also. How? For example, you have got a snake bite. You got to go to the doctor. The doctor will say that, hey, Macha, you wait, your body will produce antibodies by itself. No, doctor will directly give antibodies against the snake venom, because your body does not know about it. It is for the first time that it has been attacked, right? So basically that is artificial passive immunity. Passive because your body is not producing antibodies against it, and artificial because you are taking it from some external medium out there. Okay. And from this, by this, I have covered everything related to immunity. If it's clear, just give me a thumbs up. Hello, Kids Zone. Hello. Yes, ma'am. Got it. Perfect. Thank you so much. Some people are saying my hairstyle is so good. Thank you, ma'am. Okay. Perfect. So I hope now you will be able to do all the questions. And one more thing. Hepatitis B vaccine was produced with the help of yeast. Okay, remember that. That is also there in immunity. Okay, I know I took a lot of your time, but that's important for you guys. Okay, potato and sweet potato is what? Have edible parts which are homologous organs, have edible parts which are analogous organs, have reproductive parts which are homologous, are two species of the same genus. What do you think is the answer? What do you think is the answer? B. Tasmia says B. Harish says B. Tarun, Tasmia, Rohit, Aishwarya, Gibindra Rakshita, Rabia, and NIT. Okay, so I know there are different people out there giving me the answers. Sneha, if at all I'm missing someone's name, I'm so sorry. The answer for this question, as you are saying, it is going to be B. It is going to be B. Potato and sweet potato, Baba, what is the difference over here? Listen, this is potato. Okay, the stem of the potato goes underground and becomes enlarged over here. This is what? This is the stem. Apart from it, does it have root, Macha? Yes, Baba, it has root and everything out there. Okay, this is what, ma'am? Potato. Okay, what about sweet potato? In sweet potato, you have stem, but stem is only until here. Then you have roots, right? You have roots. Some of the roots which are there, they get enlarged. They get enlarged out there. This is what? The sweet potato. This is the sweet potato. What are they both doing? Potato, sweet potato, both of them are responsible for storage. Okay, ma'am, so you are saying the function is same? Yes, the function is definitely same. But have they evolved from the same part? No. This is stem, this is root, which is why we say that basically they are analogous organs. Analogous organs means functions are same, but anatomically they are going to be different. Okay, structurally they are going to be different. Please remember, this is one very important thing out there. Also one very important thing. Which type of evolution leads to analogous organs? Which type of evolution leads to analogous organs, guys? Yes, Sumaya, structure different. Structure different. Structure same. Sorry, anatomical structure different. Yes. Function same. Correct. Convergent. Trisha says convergent evolution. Exactly. Convergent means they were two organisms. Basically, they are converging to a point. Over here, sweet potato, do they have a choice? They are both underground. They don't have a choice. That is why they are saying doing the same function. So always remember AC, analogous organ, convergent evolution. And HD, homologous organ, divergent evolution. This is something that you need to remember. How much time asking? Please, 33 question. Manislaila. Sonakshi, can you just wait for some time? I think because we have done, if at all you could give me some time towards the end, it will be really good. Yeah? I hope you understand. Yes. So let's go ahead with the next question, guys. I hope it's okay. Right? It's okay, right? If yes, can you give me a thumbs up? If yes, we can go to the next question. Can you give me a thumbs up? Yes, divergent, homologous, exactly. And everyone who gave me the answer, guys, I'm respecting you. You are super good, okay? Perfect. Question number 35. In a plant, black seed color is dominant over white seed color. In order to find out the genotype of the black seed plant, with which of the following genotype will you cross it? So in a plant, you have black seed color, which can be this or it can be this, is dominant over what? White seed color, that is this. In order to find out the genotype of the black seed plant, which one of the plant is going to be, you are going to cross it with? Deadpool says D is the answer. PSR says D. Okay, it's okay, Saramilla. Okay, so okay. So Zainab says D. Okay, Vimla says D. Norman says D. Most of you guys are saying D, D, D. So Maya, Rabia, Muhammad Atif, PSR, Tasmia, Pavan, Deadpool, Gagana, and Rohit. Okay, so the answer for this question is going to be D. Why it is D? Listen to me carefully. Over here, it is given as black seed. The black seed can be like this. It can be like this. Both of them, because here also the dominant allele is there, it will overshadow the effect. And here, so how can you find the genotype? Listen, it looks black, so it is easier to say that it's black. But how do you find the genotype? Genotype means genetic makeup. You cannot see it, right? You cannot see it. So what do? Basically, we have to make it undergo test cross. Test cross is basically when we are undergoing, we are making sure that we cross the plant with a recessive parent plant. So over here, recessive parent plant is what? Small b, small b. Okay, recessive parent plant is small b, small b. So what do? When I cross this with this and when I cross this with this, I am going to get two different things. Let me see what is the difference. So small b over here from this parent and capital B over here from this parent. When I cross it, everyone will be capital B, small b. Everyone which is there is going to be capital B, small b. What do I understand? All of them are going to give me black seed. Okay. What about this particular condition? When I cross this with this, if at all I cross this with this, I'll find capital B, small b over here. These are the parents' alleles and these are the recessive parents' allele. When I cross this with this, capital B, small b. When I cross this with this, capital B, small b. When I cross this with this, small b, small b. When I cross this with this, again small b, no. So two of them are going to be having what? Are going to be having white seed. Two of them are going to have what? Black seed out there. So you see a different, what do you call, 1:1 ratio out there. But here all were the black, all of them were black. So what do you understand? Basically, if I get something like this, I understand that the black seeded plant is actually dominant in the genotype also. And if at all I get something like this, I understand that this is the one which is actually there. It's not pure breed, it is something like this. So what do you have to do? You have to cross with the recessive parent. This is known as test cross. This is known as test cross. Everyone who understood this, just give me a thumbs up. Okay, Markandeya. Okay, ma'am, we want quick revision. I will be doing quick revision, but if at all, Baba, you can give me some time. I will be taking another live for it, for the quick revision, so do not be worried about that. Yeah? Yes. Question number 36. Come on. ABO blood groups in human beings are controlled by the gene I. The gene I has three alleles, IA, IB, and I. Since there are three different alleles, okay, since 6 different genotypes are possible, how many phenotypes can occur? How many phenotypes can occur? Those who, though we are perfectly fast, why does this feel slow? Because we are doing a lot of explanation out there. Yeah. Make Sharda, there are live classes ongoing in a channel, Macha, please check it out. Yeah. So Saimullah says it is D, Gagan says D, Nauman says D, Pallu and Deepa and Rohit, Tasmia, Sateri, most of you guys are saying it is D is the answer. Syed Atif, right? D is what? D is 4. What is the question over here? ABO blood group in human beings are controlled by the gene I, and the alleles are IA, IB and I. Since there are 3 different alleles, 6 different genotypes are possible, how many phenotypes can occur? How many phenotypes can occur? 6 genotypes, 6 different genotypes can occur, but about phenotypes, Baba? Phenotypes, you can have A blood group, you can have B blood group, you can have AB blood group, you can have O blood group. Only 4 phenotypes are possible. Any other blood group, do you know? Ma'am, I have K blood group. Ma'am, I have O is there. Ma'am, I have L blood group. No blood groups like that. No, only 4 phenotypes are possible. So this, which is why the answer for this question is D. The question is being enlarged just to make you confused. Okay, you have been asked about phenotype, do not go and write genotype out there. Okay, D is the answer. So all of you who got D, Sri Gavi is also there, Zainab, Murthy, Revan, perfect. Melo, Hitesh, Sunil, Fiza and Trisha. I am AB blood group, is it? Okay. Ma'am, compared to yesterday, today is more active answers, right? Yes. The reason being that I think yesterday that live got stopped midway also, no? So, yes. Okay, let's go. Which is incorrect about color blindness? you have to be very sure that you read the question very carefully. Okay, which is incorrect about color blindness? This is due to defect in either red or green cone of the eye, resulting in failure to discriminate between red and green color. Is that true or false? So whenever you are reading a particular statement and a question is like this, you have to find incorrect. When you read a statement, mark it whether it's correct or incorrect. This is due to defect in either red or green cone of the eye, resulting in failure to discriminate between red and green color. Yes, this is true. So I'm writing it is true. A daughter will not normally be colorblind unless her mother is carrier and her father is colorblind. Okay, you're saying about daughter you can get only in excess condition, right? And here you are saying she will not be colorblind until and unless her mother is carrier. Her mother is a carrier means she will have only one X chromosome which is being affected, and the father is colorblind. The father is like this to be colorblind, only one X chromosome has to be affected in him, right? So when this and this comes together, there are chances that the female can be colorblind, and that is written over here. A daughter will not normally be colorblind unless the mother is a carrier and the father is colorblind. Yes, that is also true. So this is also true. By the way, I know that you guys are giving me the answers. Yes, okay. Now, if female have XCX like this, it is called carrier. But when male have XCY like this, it will be colorblind. Yes. Why? Because the female has a normal X chromosome that will be overshadowing the effect of the other chromosome out there. That is why she will be just a carrier. She will not have the disease. But here there is no one to overshadow the effect, which is why he will have the disease. So this is also true. The son of a woman who carries the gene has a 0% chance of being colorblind. What are you saying? So listen to me. If at all, I am talking about a carrier female. She has 50% chance of giving this particular gene to her son, right? 50% chance of this particular allele to be passed on to the son, and 50% of this particular allele to be passed on to the son. It depends upon luck. That's the only thing. So here it is said that it is 0% chance. So this is going to be false. So the answer for this question is going to be D. Everyone who gave me the answer as D, you guys are absolutely correct. For those of you who didn't get your answer correct, listen to my explanation. Okay? Okay, ma'am, I have applied for BSc Biotechnology. I am hopeful. Listen, biotechnology is a good field, but you have to end up somewhere really nice. Okay, and make sure you're taking it from some very good college where they have in-lab, they're giving you in-lab experience about a lot of things. Otherwise, no. Okay. Yes, perfect. So guys, if this particular question is clear to all of you, can you quickly give me a thumbs up? Everyone, can you quickly give me a thumbs up? And everyone who gave me the answer, Sunil, Navya, Sri Gavi, Hitesh, MD Suleiman, everyone, Nauman, and Melo, Zainab, Deepa, Hasina, everyone, thank you so much. And for those of you whose name I missed, I'm so sorry. Aishwarya, MD Mad Ranger, Trisha, perfect. Bhavna, Atif, okay. Cool guys, so let's go ahead with the... Yes, Kristu Jayanti is a college that you have taken. Yes, that is a good college. And yeah, one more thing, Hassan, that you can do is, you can, since I am pretty sure that you are aware about how the analytics work, okay, you know about LinkedIn and all. If you have someone, okay, just type Kristu Jayanti college and all, there will be a list of people who have alumni that would be coming up. If at all you're coming in contact with the alumni who study there, have done BSc Biotechnology, ask them, what have they ended up somewhere nice or not. That is also going to help you really well. Okay, a real-time understanding of things is going to be happening. Yeah? Yes, perfect. So let's go ahead. Question number 38. Autosomal linked recessive trait, sex-linked recessive trait, inborn metabolic error, and additional 21st chromosome. So as I always say, read the options carefully of all of them, and then over here match A first and D first. Okay, and I know that you guys are giving me, sure man, thank you so much. You're most welcome, Hassan. Okay, so yes. Most of you guys are giving me the answer as D. What is D? D is this. Okay, let me use my formula over here. Autosome linked recessive trait. Autosome linked recessive trait. It has to be sickle cell anemia or basically hemophilia. But then I have inborn hemophilia. Did I say hemophilia? I'm so sorry. Sickle cell anemia or phenylketonuria is what I meant. But I have inborn metabolic errors, so that has to go with phenylketonuria. That means this goes with sickle cell anemia. Sex-linked recessive disorder, it is going to be, I always forget the rule, sorry. So autosomal linked recessive disorder, it is going to be sickle cell anemia. Additional 21st chromosome, it is going to be Down syndrome. So A goes with 4, A goes with 4, and D goes with 1. And that is my answer. But my dear children, again, there is a more easier way. This, I feel it's complicated for the question. The easier way is, you know Down syndrome, and Down syndrome 21st chromosome is there. So D goes with 1, D goes with 1. I have already got my answer. Okay, that is also, I think you applied that. If not, then probably you can think of that. Yeah? So everyone who gave me the correct answers, guys. Saddam, good morning, dear. You're 8 minutes before. Okay, so it would be good afternoon very soon. But yeah. What about BCA? Okay, sorry, I was looking at the option over here. Bachelor of Computer Application, my dear, I have no much idea, but I've heard it's really nice. But again, I feel most of it depends upon the college and the place that you land up in and the recruitment that you have and the future plans. The BCA alone would not be enough. You would have to do up some extra courses out there which could be helping you well. If at all you are doing it at this particular point of time, you guys to have options to study with so many resources that are provided in Google. There are so many extra courses that are there. Please go ahead in your college when you get entry. Do not just stop where you have to just limit yourself to the course that you have taken, but go beyond it. Yeah? Yes. Okay, fast. Question number 39. Select the incorrect information. Select the incorrect information. Come on. BSc Nursing is a very good course. Yes, it gives you a good job, but I have to tell you, it's very draining job. Okay, I have to tell you, it's very draining job that is there. Please be ready for sleepless nights as well. But again, if at all you have been recruited outside India at some places like UK and things like that, it will be really good in terms of money. But then it's tiring job. I'm telling you that. Yes, not in India. Don't stay in India after doing BSc Nursing. Okay, so most of you guys are saying the answer for this question is going to be B. B is what? Dramapithecus, and it was more ape-like. Yes, exactly, it is wrong. So listen to me. What is the incorrect statement? Australopithecus lived in the East African grasslands? Yes, they did. Now suppose I'm not sure about East African or West African. I can leave that option for some time. Read the other option. Ramapithecus, it was more ape-like. No, Rama as in human, right? So Ramapithecus was not more ape-like. It was more man-like. Dryopithecus was more ape-like. So this is the option that is incorrect. Neanderthal man believed in burial customs. They were the first one to have a little bit brain about that, and they covered the body also. Homo habilis, the first human-like beings like hominids, that is also correct. So the only option that we are left with is going to be B. It's a very easy question. For those of you who are having doubts, please ask me in this question. If no, please give me a quick thumbs up. Okay, a quick thumbs up. Yes, someone says every job is tiring. Definitely, my job is also super tiring. But yeah, it actually depends, to be honest, in that way. But I feel that I have so many cousins of mine who have done nursing, and I feel that it pays really well and it secures your future in a very nice way if at all you end up in some government institution, especially in Gulf countries or places like that. But then again, you should be okay with the balance, that's all. Yes. Okay, guys, let's go ahead with the next question. I'm sorry, I'm not reading out your names. Now, which of the following is a correct statement? I'll read out your name for this question, okay? Now, which of the following is a correct statement? IUD once inserted need not be replaced. You are being asked about the correct statement. IUD once inserted need not be replaced? No, IUD, intrauterine device. You have to replace it. It can be replaced as well. So, this is going to be false. Government of India legalized MTP in 1981 with some strict conditions to avoid its misuse. You tell me. So most of you are saying the answer for this question is C. Hitesh and Deadpool and Sandy and Rohit, Therapist, and Sumaya, Ramya, Deadpool, Pallavi, Mahavir, Sunil, Manjula, and Srigavi, Trisha, Shraddha, Norman, Sunil, Pallavi, okay, Amulya, Faiza, Farhan. Hello Sunil. Okay, so most of you guys are saying the answer for this question is going to be C. Let me check it out. Okay, so Government of India legalized the MTP in 1981. It's wrong. It is basically 1971, not 81. Okay, so this is wrong statement. Again, false. IUD increases phagocytosis of sperms within the uterus. Intrauterine devices, yes, they increase the phagocytosis of sperm. What do you mean by phagocytosis of sperm? Basically, they take up these sperms and kill them inside. So does it happen? Yes. That is a specific feature of non-medicated IUDs. Non-medicated IUDs. For example, lipase loop. For example, lipase loop. When you're inserting lipase loop inside the uterus, it is not producing anything, but it is making your immune system very active. It is saying that immune system, hey, listen, there is something foreign over here. Tomorrow something else can also come. You have to kill it. So immune cells are super active. Now what happens when the sperm tries to enter? The immune cells goes ahead and does phagocytosis of sperms. So that can happen over here. Now D, pill suppresses gametogenesis in males. Pills suppresses gametogenesis in males. Obviously has nothing to do with gametogenesis in the case of males, because males do not take pills, right? So the only answer for this question is going to be C, and I hope my dear children have got the answer. Yes. My dream is to be IAS officer. I wish your dream come true, my dear, and give your best. Okay, guys, is it okay? If it's clear, can you give me a thumbs up? Can you give me a thumbs up? Remember, IUDs are of different types. Some mess up with the motility of the sperm, that is basically copper releasing IUD. Some mess up with the implantation, ovulation and stuff like that, or forms of mucus plug, that is hormone releasing IUD. And some does phagocytosis, that is non-medicated IUDs. Yes. Ma'am, how are you so energetic? Because of you guys. Yes. If you guys are energetic, I am also energetic. Question number 41. The edges of infundibulum possess a finger-like projections that are known as. Guys, this is going to be super easy question. The edges of infundibulum possesses finger-like projections. That is what? That is going to be what? Are the site of fertilization, help in collection of ovum after ovulation, are responsible for release of egg? None of this. Okay, ma'am is Malayali. Yes, I'm Malayali. Sanjay says B is the answer. Hitesh says B. Tarun B. Econ MBB Sita. Okay. Manjula says B. Mahavir says B. Rohit B. Vandana and Iron Tom. Okay, so Maya, Rohit, Sandy, and Mansina, Khushi. Okay, so my dear children, Noha, Gazi, Trisha, everyone, most of you guys are saying it is B. And what is B? B is going to be help in the collection of ovum after ovulation. Exactly. So if at all you see at the oviduct, okay, this is the infundibulum and this is the finger-like projection, that is the fimbriae. You call this as what? Fimbriae. It basically, this is the ovary that is there, and it goes and touches the ovary and says, Macha, if you have some egg, give me, I will collect it and give it back. So basically, it helps in collection of ovum after ovulation. That is the logic. So the answer for this question is going to be B. Very easy question, right? Clear? Let's go ahead. I'm not asking you to give me thumbs up because I think this is an easy question. Question number 42. You can write this one. During spermatogenesis, which cells are the first to contain haploid number of chromosomes? During I initially in my second PUC first quarterly failed, but in finals I got 70% biology. Thanks, Jasmine Ma'am. Oh, that's so sweet. Wishing you the best, dear. Okay, so during spermatogenesis, Baba, which are the cells that has haploid number of chromosomes? Any idea? B. Some people are saying C. Some people are B. Guys, give me one option. You are giving me A, B, C, D option. It's like MCQs for me. You have to choose. C. Okay, so most of you guys are saying it is secondary spermatocyte. Always remember, easy trick. Haploid. When the term haploid comes, every time it has to be something that is secondary. It can be secondary spermatocyte or secondary oocyte. From that point onwards only you get haploid. Before that it was always diploid. Okay, and then finally the ovum and the egg is also, ovum and the sperm is also haploid, but the first point is spermatocyte. So over here you have got the primary spermatocyte. It undergoes division. You got the secondary spermatocyte. It was 2N over here. It undergoes meiosis 1, and you know meiosis 1 is reductional division, so you get N out of it. So basically what do you get? You get N over here. Finally again it undergoes division. Okay, this is meiosis 2. At the end of meiosis 2, you do not reduce the number of chromosomes. It remains the same. Okay, it remains the same, which is why it is N only. So this is secondary spermatocyte. This is where for the first time you are going to see haploid content, and basically then you have the spermatids, which are also haploid in nature. Okay, so the answer for this question is going to be C. Again, easy question. Guys, if it's clear, can you quickly give me a thumbs up? If it's clear, can you quickly give me a thumbs up? Tarun, I strictly think you should be asking that to Ankit sir. If at all you have backlog for rapid revision, I will not be a good person to talk about that. Okay. if at all you know the list of chapters, I have provided you the list of chapters. If you can, please refer to those chapters first, if at all they are of high importance. Okay, don't miss out on those chapters. But the way of how you can deal with that, I think Ankit sir's video could help you well. Watch that video on Ankit's of KCET fixed math 45 plus. So in that, I think he has said something, okay, which could be helping you well. Yes, perfect. Question number 43. David Tillman's long-term ecosystem experiment using outdoor plots showed that. David Tillman's experiment, my dear children, what did it show? Increased diversity contributed to lower productivity. Increased diversity contributed to higher productivity. Increased diversity have no impact on productivity. Increased diversity have unpredictable impact on productivity. What do you think is the answer for this particular question? Hello, Sunil. Hello. I'm seeing you, dear. Hitesh says B, Roman says B, Sharda says it is C. Okay, Iron Tom says B, Tarun says B, and Shankar says B. King is there. Hello, King. Norman, Uday, most of you, Komali and Sri Gavi, Gagana, Trisha, Preetham, thank you so much, dear Preetham. Manjula, Shivalinga. Okay, so B is the answer for this question, is it? Yes. So David Tillman, Baba, he was the person who said that if at all you're increasing diversity, that is obviously going to increase the productivity. If you talk about multiple plants out there, what is going to happen? These plants, suppose it can be big, this can be small. Anyway, sunlight can be utilized in different levels out there, and basically all of them can contribute to productivity, higher productivity. So that is what he was talking about. And not just productivity, he also spoke about stability. Stability as in, for example, I am an organism. Suppose I do not have this particular plant. This particular plant is somehow dying. Another plant is there. I have an option to go with the next one, right? You get it? So that means it is the stability of the ecosystem is being increased with the options that is being given to us. Yeah, we can still survive. Yes. So the answer for this question is going to be B. Other options are wrong. Why? Because here lower productivity is being mentioned. That is obviously not true. Increased diversity have no impact on productivity. That is also not true. Increased diversity have unpredictable. That is also not true. So the answer would be B. Cool. Cool. Not till every day, ma'am. I'm from Wayanad. Okay. Question number 44. I hope that's clear. Guys, if at all it's clear, can you just quickly give me a thumbs up? My head is spinning now. Okay. Now, ecosystem chapter tough to understand. Is it? Ecosystem is actually an easy chapter, but you have to just go through it. It's an easy chapter, trust me. Oh, my aunt is from my aunt. Is it? Okay. So, yes. Perfect. Sangeeta, what's the problem, Acha? Guys, by the way, if at all you feel that your doubt is remaining as a doubt, please stay back until the end of the session. I'll be clearing it. Okay? Yes. Next. When more than one adaptive radiations appear to have occurred in an isolated geographical area, it is called. When more than one adaptive radiations have occurred in an isolated geographical area, what do you call this? Divergent evolution, convergent evolution, anthropogenic evolution, saltation. Okay, Queen says B is the answer. Hitesh says C, Gagan, Trisha, and therapist. Most of you guys, Panvit, most of you, Navya, PSR, Sumaya, and Sharnya. Okay, so most of you guys are saying B is the answer, and when you say B is the answer, we go with convergent evolution. Yes, so listen to me, for those of you who are not understanding. I have a group of organisms right over here. Okay. It was there in the middle, but some felt that if at all I move here, I am going to live better. So some went there and they were actually able to live better. Okay, probably they might have had some changes. Okay, since when they moved up there, they were able to explore things better out there. You might have heard about Darwin's finches. You know what? Darwin's finches, which are over here, they had some mutation. So when some of them moved to other area, they were, in fact, with the different type of beak that they were having, they were able to survive there. Right? So they got adapted there. This is known as adaptive radiation. Now here it has been said that basically if one group of organism is doing this, I'll say it is divergent. It is going to be divergent. You had a common ancestor, they are diverging to different habitats. But Baba, when you're talking about multiple organisms, okay, when you're talking about multiple group of organisms out there over here, they are all converging. they are all moving, right? They are of different ancestors, but now they are all together over here, right? So what do you mean by that? Convergent. They are converging to a point, to a single point. So when multiple adaptive gradations occur, we say it leads to convergent evolution. So they might be of different places, ancestors, but yes, this is the thing. Okay, guys, please restrict yourself to talking things that is there over here rather than chattering amongst yourself. If at all you have understood, give me a thumbs up. Thank you so much. Some people have already started to give me thumbs up. They already know that is the next question. Okay, question number 45. Question number 45. SMAC is chemically dash, which is white and odorless and crystalline in nature. This is obtained by dash. Here A and B refers to what? Here A and B refers to what? Any idea? SMAC is chemically dash. What do you call SMAC as? Mahavi says B is the answer. Rohit says B. Okay. Panvit A. Sandy A. Somaya B. Okay. So guys, the answer for this. So A and B is something that you're confused with. Listen, you have morphine and you get heroin. SMAC is what? SMAC is heroin. SMAC is heroin. Okay. Morphine, which was there, it undergoes a process that is known as acetylation, and that's how you get SMAC. Heroin, or the other term for it is diacetylmorphine. Diacetylmorphine. So SMAC is chemically what? Diacetylmorphine, that is given over here. Fine, which is white and odorless and colorless and basically crystalline in nature. This is obtained by what? Acetylation of morphine, which is why the answer for this question is going to be A. Okay, got it? Cool? Clear? So everyone who got this particular question, give me a thumbs up. Everyone who got this question, give me a thumbs up. Quick thumbs up, come on. Perfect. Super cool. Yes, got that. And by the way, everyone who have not hit on the subscribe button or not on the like button, do that right now, okay? So choose the incorrect statement. You have to choose the incorrect statement, and it goes on like this. Both cartilaginous and bony fishes are dioecious. Dioecious means what? You have to have both this. Sorry, dioecious means the sexes are going to be separate. Okay, cartilaginous fishes show sexual dimorphism. That means males and females would be looking different. Yeah, that is known as sexual dimorphism. Male cartilaginous fishes have claspers. Female cartilaginous fishes have claspers. What do you think the answer is? It is incorrect. Ma'am, Neha bin. Ma'am, previous year question matram nokia madiyo. Padilla. You have to go ahead. So it's not just previous year questions. You have to go ahead with other questions also. But PYQs are unavoidable. You have to look at it because you will get an understanding of the important concepts. But yes, you have to look for the other questions also. So first PYQs, then extra questions, both of them. Okay? Yes. So most of you guys are saying the answer for this question is D. D is what? D, E. I went, look now, my brain is not braining. I went to tag that as D. This is D. Female cartilaginous fishes have claspers. Baba, claspers are there for what? Claspers are there for transfer of what do you call, the gametes out there. Or basically it is going to help in the case of sexual reproduction for the males to pass their gametes. Okay, it's not found in the case of females, it is found in the case of males. So here it is written female cartilaginous fishes have claspers. Wrong. It is going to be absolutely wrong. Okay, so D is the answer for this particular question. Who you, ma'am? What all questions are come? Ma'am, can you sing? Ma'am, you need sleep? Ma'am, you are sweet? Oh my God. Thank you so much for telling the nice things, and for people who are saying, ma'am, you need sleep. Definitely, I agree with you. I need sleep. Okay. So yes, is this question clear? Ma'am, you pretty. Thank you so much, guys. If it is clear, can you quickly? I'm saying quickly. Give me a thumbs up. Okay, quickly. Can you give me a thumbs up? Yes, perfect. Ma'am, +1, +2, +2. 40 questions are going to come from your PU2, and 20 questions usually come from your PU1. And if you join the WhatsApp channel, you will get the PDFs out there of the most important series that has been done. Okay? Yes. Okay. Consider the following statement. External ears, this is from PU1 guys, come on, concentrate. External ears are absent in frog, only tympanum with internal ears aids in hearing. The eyes of frog possess single unit, hence are simple. Hence are simple. What do you think is the answer for this? A, B, C, D options are given to you. You have to choose from this. Deadpool says the answer for this question is A, okay, and Panvit says A, Somaya says A, Deadpool says A, Hitesh says B is the answer, huh? Okay. Queen says A, Amulya A, Somaya, Shankar, Panvit, no ma'am, can you explain once more? Sangeeta, yes, I will. Just give me a second, okay? I'll just after this. Pallavi, okay, so most of you guys are saying A, some people are saying B is the answer. Why is that so? Okay, so that means we have to check. Listen. External ears, this is the external ear. Do we have it? Yes, we clearly have it. What do we call this? Pinna is what you call it, yeah? This is absent in the case of frog. Have you seen a frog with ears? It would be really funny to spot that. So they do not have the ears, okay? They only have the tympanum, that is the eardrum and everything, and the internal ear out there, okay? That is there for earring. This is correct. Now the eyes of frogs process single unit, hence they are simple. Yes, that is also correct. They have a simple eye. They have only single unit, it has a simple eye. So basically, can I say both the statements are true? One is true, so the answer should be both A and 2 are true. This should be the answer. A should be the answer. So for those of you, 2 is false. Why is 2 false? Both are true, okay? Both are going to be true out there. And just give me a second. Okay. Guys, I need a quick second over here. Someone was getting confused with this particular topic. Choose the incorrect statement is being asked. Rest every statement is correct. Cartilaginous and bony fishes are dioecious. Dioecious means they have 2 separate sexes. Male and females are separate, right? Yes. Have you seen a fish having both the male and female part? No. So it is going to be dioecious. Cartilaginous fish shows sexual dimorphism. Again, over here it is clearly visible. The males are different and the females are going to be built different. They will have some changes in them. Now the male cartilaginous fishes have claspers. Do they have claspers? Yes. The claspers are something that is useful for transferring the gametes out there. Female cartilaginous fishes, do they have claspers? No, that is not in them. That is only a feature found in the case of males. Okay, I hope that is clear. Okay, now someone was asking me about complex and simple eyes. Complex means basically there would be multiple units within your eyes as well, which is going to trap the light and which is going to make things much clearer. But over here in the case of simple, they do not have too many things are there. Tamil puriyama, konjam puriyam. Okay, so yes, question number 48. Guys, I hope this is clear, right? Is this clear? If yes, can you give me a thumbs up? Guys, thumbs up. Don't get misleaded. Okay, yes. Next question, question number 48. Alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gum, spices. How many of them are primary metabolites? Any idea how many of them are going to be primary metabolites? Any idea? B. Mahesh says the answer for this question is D. Queen says it is D. Prajwal says it is D. Sandy says it is B. Why B? Okay, let me check it out. Okay, Nagavini says it is D, Nauman says it is D, and so Sunil says no. Okay, but most of you guys with what I'm seeing are saying it is D. It is D. Some people are having no idea about it. It's okay. Okay, it's okay. Let's check it out. So all these things that are being mentioned over here, none of them are primary metabolites. Okay, all of them are going to be secondary metabolites. Whether it is alkaloids, whether it's flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums and spices, all of them are secondary metabolites. All of them have different functions. For example, you talk about quinine, you talk about morphine, we spoke about it, right? They are basically alkaloids. They are there for defense. Flavonoids which are there, they are basically pigments and antioxidants. They are not the ones essential for growth. So there are multiple functions are there. You just need to remember that they are not the primary ones. They are basically secondary metabolites out there. Cool? So D is the answer for the particular question. Is this clear? All of this are straight questions. They are not what you call complicated questions out there. There is no explanation that I can give for that, right? Muhammad Atis has a nice way for giving D. That's cool, clear, perfect. Guys, thumbs up needed. Without thumbs up we cannot go ahead. Clear, ma'am, easy peasy. Yes, perfect. Hey, okay, ma'am, I meant to say no. It's okay. So which of the following resembles with normal mitosis, Baba? Meiosis 1, meiosis 2, both or none of this? Both or none of this? Meiosis 1, meiosis 2, both or none of this? Which of them resembles with normal mitosis? What do you think is the answer for this particular question? Cool. Okay. So Sandy says it is C, Bhavish says C, Deadpool says B, Rakshita says B, Queen says B, okay, Amulya B, and oh my god, that's going, Rakshita and Norman, Aditi is also there, Zainab is also there, Iron Tom is also there. So the answer for this question is very simple. It's going to be meiosis 2. Listen, what happens in mitosis? For example, I start with 46 number of chromosomes. I do mitosis. There are 2 cells out there. It will be same, 46 number of chromosomes will be there. This is why I call this mitosis, and it's an equational division. I call this equational division. Why? Because the number of chromosomes over here and in the parent cell is going to be the same. Now what about meiosis? In meiosis we have studied one very important thing. We start with 46 number of chromosome. Meiosis happens in 2 stages. Okay, 2 stages. So meiosis 1 is there when you get 2 cells. It will have half the number of chromosomes. Half the number of 23. That is why you call this as reductional division. Ma'am, is meiosis over by this? No. There is one more division that takes place. So do you think it will get more? Once more it will get halved? No. It remains the same. It becomes 23. So over here, if at all you see, it's like an mitosis only. It's like mitosis only. Why? Because the parent cell number of chromosome and the daughter number of chromosomes remains the same. But this is meiosis 2 that happens, and it has high resemblance to mitosis. So the answer for this question is going to be B. Okay. Bharati, yes dear, tell me. Zainab, I want to talk with you only for study. Baki kuch nahi. Oh my god. Okay. So guys, ma'am repeat. Okay, ma'am will repeat. Listen. In the case of mitosis, suppose I get a wound over here. Okay, I get a wound over here. What type of division happens over here? Mitosis. I just want to replace it with other cells which are of the same type, right? So if at all I have a cell with 46 number of chromosome, it is going to divide. It is going to form 2 cells with the same number of chromosome. That is known as mitosis. That happens in all our general cells out there. You call these cells as what? Somatic cells. Okay. Now, talking about meiosis. Meiosis happens only in gametes. For example, you only think, if the sperm which is there, it carries 46 number of chromosome, and the egg also carries 46 number of chromosome, 46 comes together, it becomes 92 number of chromosome. That is very different from the normal number of chromosomes that we have. We always have 46 number of chromosomes. So what is the best way to make it 46 again? Half it. So half comes from sperm, half comes from egg, and basically 23, it becomes 46 again. So this is there for meiosis is there for what? For the gametic cells only. And what happens in meiosis? 2 stages. In the first stage, 46 number of chromosomes which were there, it gets reduced to half. Both the cells will get only half the number of chromosomes, 23. Then there is meiosis 2. that's when we say that meiosis is completed. So meiosis 2 happens when this cell again is divided into 2, this cell again is divided into 2. If at all you see over here, does the 23 becomes reduced over again? No, Baba. It remains the same. 23 remains the same over here. So we say it is equational division that is happening over here. If at all you see there, the number of chromosomes was same over here. If at all you see here, the number of chromosomes are same over here as well. So don't you think it is resembling like mitosis? Yes, Baba. It resembles like mitosis. Cool? Yes. Hiya. Clear na? Okay, next question. Oh my God. Okay. Question number 50. Read the following 4 statements. External nostrils leads to nasal chambers through the nasal passage. External nostrils leads to nasal chamber through the nasal passage are there. Is this correct or not? You have to find what is true. So please start marking from here also. It is true. External nostrils which are there, it takes the nasal passage are there. Yeah. Now, nasopharynx is a portion of pharynx. Yes, nasopharynx is a portion of pharynx. Pharynx is a common passage for food and air. Larynx is a cartilaginous box with the help of which you get the voice production. True. All of them are true. What do you think is the answer for this particular question? 4. 4 is the answer for this question. And for those of you who are not understanding, just give me 1 second. Okay, not 1 second, 1 minute. This is Pappu. Okay. And over here, you will be having the nasal passage. Okay, this is the external nostrils through this nasal passage. And from here, Baba, you are going to get the buccal cavity opening. Both of them are going to end up over here. What do you call this place as? This is pharynx. So don't you think whatever food you eat, for example, I am eating noodles. So noodles basically are going to pass through this only. Similarly, Baba, the air that you breathe, that is also going to be passing through this only. This place of the pharynx, which ends up over here, this is known as nasopharynx. Okay. And then this also has a different name over here. All of them basically are going to be there is a common spot that is known as pharynx, and this pharynx, Baba, later is going to divide into what? The windpipe and the food pipe. That is the esophagus. That is the logic out there. And with this, you got the answer. Got it? So basically all of them are correct, and in the windpipe you will be finding larynx, which is a cartilaginous box. Please make sure you read the options carefully. Why? Because sometimes they can give you a bony box also. At that time you should be able to give the answer, right? Clear? Pappu again. Yes. Khudre vittam Pappu. Yes, exactly. Guys, just give me a second. Okay? Hello? Hello? Nan live illa. Live edukko. Nan pinna vilcha madhyo. Okay, bye. Okay. Ma'am, repeat. Ayyo. Repeat here. Listen, this is nose, this is mouth. Yeah. From here your food goes, from here your air goes. The common passage where it reaches is known as pharynx. The end of the nasal cavity where it opens into the pharynx is known as nasopharynx. Okay. And over here, Baba, the pharynx leads to what? The food pipe and the windpipe. So basically all these options are correct, and larynx has not been mentioned over here, but I'm telling you it's there in the windpipe and it is cartilaginous in nature. Okay. Cool. Next one. Next one, question number 51. Is this clear? Guys, I am tired actually. I've been standing from 9:30. My back is paining. Okay. First cardiac sound is associated with. Come on, just 9 questions. First cardiac sound is associated with what? Hitesh says A, okay. Vandana says B and A, okay. Lisha says A, okay. Lisha is there. Hello, Lisha. And Deadpool says A. Panvit says A. Kyun, Sumaya, Iron, Lubdub. Yes. So most of you guys are saying A. Trisha, Arif, Bharati, Sumaya, Prajwal, Ramya. A is what? Tricuspid and bicuspid valve. Exactly. So listen to me very carefully. You have the right atrium over here. You have the left atrium over here. You have the right ventricle over here. You have the left ventricle over here. Okay, now between the artery and the ventricle, obviously you are going to have what? You are going to have a valve. Why valve? Suppose the blood is filled over here. Imagine, I'm not going to repeat it. Listen to me carefully. The blood is filled in the atrium. What is going to happen? They are going to contract. When they contract, what is going to happen? All this blood which is there over here in the atrium is going to move to the ventricles, right? Now the ventricles have the blood. Ventricles, what will they do with the blood? Basically, they have to transport this blood to the rest of the body parts. The left ventricle transport to the rest of the body part. The right ventricle transport it to the lungs. That is the logic. Okay, that is how it goes. For transferring, obviously, these guys have to undergo what? Contraction. Now, when they contract, you use your imagination. some amount of blood will be going, okay, when they contract, what is going to happen over here? Some amount of blood which is there, it is going to go up. When you contract things, pling, things are going to go up. When things are going to go up, it can go to the right atrium. That means we have to stop that. For stopping that, what do we need to do? We need to close this valve, my dear children. We have to close this valve, right? And that closure of the valve which is there is sounding like lub. It is sounding like lub. What do you call this valve? Always remember, between the right atrium and the right ventricle, you have the tricuspid valve. Tricuspid valve. So I remember it as RI. You know, you have RI, so RI for right. So in the right side, you have tricuspid valve. Over here, you have what? Bicuspid valve. Bicuspid valve is also known as mitral valve. So the closure of this is sounding like lub out there. Cool. Cool. And why it is known as tricuspid? Because it has a cusp like this. There are three things which are there. They close up like this. Three flaps are there. Okay. Works na macha. Okay. Lub, and the next one is dub. Yes, the dub one which is there, Baba, the dub is coming because of the closure of the semilunar valves. That means over here also, between the ventricle and the arteries which are going to be there, which are going to carry the blood. So when the blood contracts, to prevent the backflow, you close this. Okay. Now when the blood contracts, you are sending it to the artery. From the artery also the blood can come back. To stop that, you have certain valves over here. These valves are known as semilunar valves. So closure of that is known as dub. So lub, dub. Cool? Ma'am, we love you. Love you too. Okay. So guys, is it okay? And Sunil, OA, do not spam over here, guys. If it's okay, can you just give me a thumbs up, guys? If this question is clear and you will not forget, just give me thumbs up. Thumbs up, fast. Yes, perfect. Cool. Children, I am just shifting to a question because I think it will make more sense right now. And where is that question? Yes, this one. Try out this question. Identify A in the below figure. A in the below figure. Don't worry, I'll cover the other questions also, but I'm just telling you because this will make sense now. A is what? So A is some strand-like things that is over here. What is A? That is the question. Any idea? Trisha says the answer for this question is C. Elisha says it is C. Okay. Nauman says it is B. Manjula says it is B. Sri Gavi says it is C. Nauman says B. Guys. Thank you, Shobraj. And so the answer for this question, Harish is saying B. Guys, I am asking about this thing. This is not a valve for sure. There are some strand-like things which are being mentioned over here. What is this? Chordae tendineae, exactly, therapist. So those of you who gave me the answer as B, you guys are correct. Okay, B is correct. Listen to me very carefully. So the thing is, you have to use your imagination, okay? So chordae tendineae is right over here. There are some string-like things that is there. And basically, they are attached to what? They are attached to the papillary muscles of your ventricles. So basically, use your imagination. You have your ventricles. You have your papillary muscles right over here in the ventricles. You have some strands. Okay, and these strands are attaching to what? To basically your valve out there. So this is the valve. Okay, now use your imagination because imagination is very much important over here. Atria pump the blood, now it is there in the ventricles. Now, when the ventricles pump the blood, what is going to happen? The blood goes up. When the blood goes up, the flaps which were like this, they close. But understand there is too much pressure such that the flap can open inwards. The flap which is there, it can open inwards also. So you have to make sure it does not open inwards. It stays at that particular location. And that is the use of this chordae tendineae. So chordae tendineae makes sure that this particular things are attached to the muscles of the papillary muscles of the ventricles and make sure it just stays there and does not close up. Got it? So that is the logic. Okay, so that is chordae tendineae. Push, yes, tish. So push, tish. I know that you like the voices, but yeah. Yes, exactly. They act like strings that hold the valves in place during heart contraction. Okay, so over here you will be having, so the picture is not really good, but I'm just telling you. So you will be having your tricuspid valve over here. Okay, these are the chordae tendineae and it will be attached to the papillary muscles. So when the ventricles contract, basically the flap which is there, it will be not just closed, it can open up, which is why the chordae tendineae pushes, make sure that it stays there only to a limit. Fine. So the answer for this question is B. If this question is clear, everyone, can you give me a thumbs up? Ma'am, are you certain about? I have not watched any of the animes and I am being very honest. Okay, not because I have not explored it actually. So sorry. Yes. Okay. So now I'm going to the questions which were already there. Yes. Okay. Question number 52. Which of the following statement is correct? The ascending limb of loop of Henle is impermeable to water. The descending limb of loop of Henle is impermeable to water. The ascending limb of loop of Henle is permeable to water. The descending limb of loop of Henle is permeable to electrolytes. What do you think is the answer for this particular question? Come on. Ma'am, your class understanding, I am 100% 60 out of 60. Is it? Thank you so much. You will get 60 out of 60. Keep on going, okay? Sagar, thank you so much. Okay, so most of you guys are saying the answer for this question is A. Deadpool, Vidya, Zainab, Lahin, Sandy, Purvita. Okay. Perfect. So the answer for this question is A, is it? Let me check. So the loop of Henle, Baba, if at all you have seen, there is descending loop of Henle right over here and there is ascending loop of Henle, right? There is ascending loop of Henle. So here it is kind of thicker and here it will be thinner. Anyway, so this is descending, this is ascending. Always remember, descending which is there, it is going to be permeable to water. Permeable to water, and it is impermeable. Impermeable, that means it will not allow electrolytes to pass through. It is only allowing the water to pass through, it is not allowing the electrolytes to pass through. What about ascending loop of Henle? It's the opposite. It is impermeable to water. Impermeable to water. Does not allow water to go, but it is permeable to what? Permeable to the electrolytes out there. This is very much important for maintaining the counter current thing. Okay, so over here, the ascending limb of loop of Henle is impermeable to water. Is it impermeable to water? Yes, Baba, it is impermeable to water. Okay, and that is the only thing that is correct. Descending limb of loop of Henle is impermeable to water. No, it is permeable to water. So this is wrong. Ascending limb of loop is permeable to water. No, Baba, it is impermeable. No, that is what we studied. This is wrong. The ascending loop of Henle is permeable to electrolytes. No, it is impermeable out there. So basically, the answer for this question is going to be A. This is very important. If it's okay, if it's clear, can you give me a thumbs up? Ma'am, like this question is only coming in KCET. Yes, yes, exactly. Questions could be coming in KCET like this. So, yes. Perfect. So please remember it. Do not forget it. This side, please remember blue color. Descending, you remember blue color. That's all. You will remember everything else. Yes, it's an electrolyte, sodium. Okay. Question number 53. Myofibrils appear striated due to the presence of myofibrils, which are there. They are found in the case of muscles. I hope you know that. And they appear striated. Why do you see dark band, light band, dark band, line band? Why do you think there's a striation out there? Actin in the lighter region and myosin in the darker region. That's why. Actin throughout the length of the myofibril. Myosin in the lighter region and actin in the darker region. Myosin throughout the length of the myofibril. What do you think is the answer for this particular question? What do you think? Sandy says the answer for this question is going to be D. And Ramya, Aaron, thank you so much. Deadpool says it is A. Kishan says it's D. Panvit, guys, are you reading the options carefully? Panvit says it is A. Vidya says D. Okay, Amlu says A. Lisha says A. Queen says A. Okay, Shalam says A. And Bhuvan, hi, Amua. Who's Amu? Okay, Aaron, Panvit, Maya, Trisha. Okay, Black Man. Arshia. So guys, Ramya, so the answer is actually A only. If at all you look at the myofibrils, myofibrils are made up of what? Sarcomere unit. Yeah, and the sarcomere unit is made up of what? These thick bands which are known as what? Myosin. And there are thin bands which are made up of actin filaments. So thin actin, myosin is going to be thick filaments. Okay, always remember. Now how do you remember it's thick? So myosin, basically if at all, let's see. I do not know if it makes logic or not. Myosin and okay, so there is one letter more, so basically this one is the thicker one. Okay, this one letter is less, so this one is the thinner one. You can remember this. This picture is very important. You have got so many questions based on this. Yesterday day before yesterday, while I was solving a PYQ, you saw the question, right? So this is very, very important. This is one of my favorite chapters, is it? Okay, exactly. So guys, please remember, I hope I do not have to explain this. If you have any confusion regarding sarcomere, please make sure you see my previous life. Yeah, so actin in the lighter region and myosin in the darker region is the answer. Actin is not present throughout the length, that is why it is wrong. Myosin in the lighter region would be wrong. Myosin throughout the length, that also would be wrong. Ma'am, Amalu, I have never forgotten you, dear. I still remember you. Don't think like that. Okay? Guys, 54th question. The forebrain consists of, very easy question. Come on, come on, come on. The forebrain consists of what? The forebrain which is there, it consists of what? Cerebellum, thalamus, hypothalamus, pons, cerebrum, medulla, amygdala, hippocampus and brainstem, and thalamus, hypothalamus and cerebrum. Okay, so most of you guys are saying it is D. Deadpool, Sri Gaurav, Lisha, Pooja is Black, Panvit, Vidya, Nauman, Bharati, okay, Ram Charan, Hitesh, okay. B Reddy is also there, Iron is also there. Okay, so guys, what is the answer for this question? Lehin, Nandita, Mukund. So the answer for this question is going to be D. Why not A? Because here cerebellum is mentioned. Cerebellum is a part of your hindbrain, not your midbrain. Sorry, not your forebrain. So this is wrong. Pons, cerebrum, medulla, again it's wrong thing. Pons which is there, medulla which is there, a part of your hindbrain. Amygdala, hippocampus and brainstem, again not the thing. Thalamus, hypothalamus, cerebrum is the only answer for this particular question. Your forebrain consists of this. Okay, I'll tell you a basic funda to remember this. I mean, it's not a funda, it's something that we study in 10th grade also. So forebrain is the largest part of your brain. Right ma'am. It contains cerebrum. The larger part of your brain is required for what? For thinking. For thinking, you're doing that right now. For holding your pen, for typing, what all things that you're doing with the help of thinking and all, voluntary actions that you're doing is controlled by the largest part of your brain, which is known as cerebellum. Sorry, cerebrum. Sorry, not cerebellum, cerebrum. Now, hypothalamus is the one that is responsible for all the physiological function. For example, I am thirsty, I am hungry. All these things, my sleep drive, everything which is there is being controlled with the help of what? Hypothalamus, not sleep drive. Okay, anyways. So, hypothalamus is the one that controls it. Thalamus and hypothalamus coordinatingly. Now, Baba, you have midbrain. The midbrain's normal function is it relays. Whatever information is there, it helps in passing through. Now the next thing that is there is the hindbrain, which is the next important part. For the hindbrain, you have the second largest part of your brain, that is your cerebellum. And cerebellum, the easiest way to remember it, the L over here looks like the straight line, right? So cerebellum is responsible for our posture and balance. It maintains our posture and balance. So people who drink, okay, they drink alcohol, is the cerebellum that gets affected. That is why they go like this. That is a region. They do not have the posture and the balance. Now you have the brainstem, which is made up of what? Pons, as well as what? As well as the most important part of your brain, medulla oblongata. Medulla oblongata is responsible for maintaining your heartbeat, blood pressure, everything, respiratory functions, everything. And for coordinating with that, you have pons as well. So please remember, the most important part lies over here. And yes, cerebrum is also important, definitely, but I'm just telling you. Yes? What is happening? Jum, 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 jum. Guys, what's happening over here? Which part of head is there for dreaming? That is a homework for you guys. Okay, so which is incorrect about thyroid hormones? Come on, guys. Come on, come on, come on. Which one is going to be the incorrect statement about thyroid hormones? Guys, to be honest, I am very tired. My back is really paining so bad. So I'm also trying to finish it as soon as possible. But yes, Deadpool says D is the answer. Uday is also saying, okay. Uday has no answer. Why is it coming like that? Okay. Lisha, Panvit, Meaningful, Shri Gaurav, Vidya, Be Ready, all of you, most of you are saying D is the answer for this question. Is it D? Let me check it out. Thyroid hormones, that is T3, that is triiodothyronine and thyroxine, are synthesized by the follicular cells? Yes, they are synthesized by the follicular cells. Correct. Thyroid hormones support the process of red blood cell formation? Yes. They help in erythropoiesis. Listen, it happens in the kidney, red blood cell formation happens in the kidney with the help of erythropoietin, but yes, thyroid hormones are going to help in that, okay? Now, thyroid hormones control the metabolism of carbohydrates, proteins and fats. Major function of thyroid hormones is to control your basal metabolic rate, so obviously it is going to control the metabolism of all these things. That is also correct. Now, this is incorrect. Why? Maintenance of water and electrolyte balance is not influenced by the thyroid hormones. Wrong. It is influenced by the thyroid hormones. It goes and influences the kidney and says that you should not be taking away the electrolytes or the water out there, so it maintains the electrolytes, okay? So that would be very wrong to say that. So this is the statement which is going to be wrong. Rest everything is going to be correct. If this particular concept is clear to you, can you give me a thumbs up? Okay, can you give me a thumbs up, everyone? Yes, perfect. Ma'am, drink some water, it's summer, iron. Yes, dear, I have to. Yes, I'll. Thank you for reminding me. Okay, perfect. So, next question, very easy question. Identify the vertebrate group of animals characterized by crop and gizzard in their digestive system. So, who has crop and gizzard in their digestive system? Any idea? And for children who are spamming, please be aware, okay, I will not be like this if at all you make nuisance in the class, okay, so be aware of that. And one person I've already put on timeout, okay. Deadpool says A, Navman says D, and Pandit says A, okay. Nagavani says it is C. Why dear children have different answers? Muhammad Saqib is there, Shobharaj is there. Most of you I think are going with A. Are going with A. Some people had options like C also, some has B also. Baba, what is the answer for this particular question? What is the answer for this question? Crop and gizzard, whenever this thing comes up, no, just understand it is going to be aves. Aves as in birds, Baba. And I'll tell you one very important fact about them. Just let me drink water, one sip of water, okay? Okay, the fact about them, Ostithis, Baba, it's bony fishes. Why will, okay. Anyways, so over here, in the case of aves, they take in the food, that is the birds, they take in the food, they store the food in the crop. That is the first part that the digestive system goes into. So crop is where their food is going to be stored. Now it went to the first stomach that is known as proventriculus, where actually the digestion and everything happens. Okay, then it goes to the ventriculus, where, my dear children, which is known as gizzard as well, where finally things are going to be broken down mechanically. And how are these things broken down? Basically, the surface of the gizzard, no, it will be having some tiny spike like things like this, which is going to mechanically break them down. And if at all you have seen birds, while they peck things, peck, they also take some grits and pebbles, small grits and pebbles along with their food. Why? Because it goes to their gizzard, and in the gizzard, Baba, this pebbles or basically grit which is there, it helps in breaking down the food. It helps in breaking down the food. So please, if at all you have noticed it, you might have, you might feel it's like really crazy thing. Yes. So the answer for this particular question is going to be A. Cool. Clear? No? If it is clear, can you quickly give me a thumbs up, guys? Yes, it is. Okay, Varidraj, hats off to you, ma'am, you're teaching. So that's so sweet of you. Thank you. Okay. Cool. 3 more to go. Let's go. And by the way, not 3 more to go. One we have already covered before. Chordate tendinae, remember? We have covered. Only 2 more to go. Okay. So, question number 58. In the given list, how many hormones are included in steroid hormones? Insulin, cortisol, estradiol, glucagon, and progesterone. What do you think is the answer for this particular question? Any idea what is the answer for this particular question? In the given hormones out there, you have to tell me which one, how many of them are going to be steroid hormones. Any idea? D. Different people have different answers. Some people, cockroach does not come in Aves, it comes in arthropoda. The answer for this question is going to be 3. It is going to be D. And can anyone tell me what are the hormones which are included in steroid group? Which are the hormones which are there in the steroid hormones? Any idea? Can you please tell me? It will be really great if at all you could tell me. And by the way, just give me a time. You. Insulin, glucagon and progesterone. Okay. Progesterone, cortisol and estradiol. Okay. And that was Iron Tom who said that, right? So guys, the answer for that is, if at all you are being asked about the steroid hormones, that is cortisol and estradiol and also progesterone. Okay. This and this. These 3 are the ones which are steroid hormones. Okay, so please know about that as well. Very important. Cool. Now, I hope that question is also clear. We can go ahead. And one of the side effects of the use of anabolic steroids, in females. Guys, this is important. This is for, this is not for, what do you say? This is not of PU1, this is of PU2. Come on, try to attempt it. One of the side effects of the use of anabolic steroids in the females is what? Lisha says the answer for this question is C. Nagavini says it is C. Deadpool says C. Panvit says C. Okay. Sakib says D. Shama, Sri Ganesh, C. Okay. Preeti says C. Most of you guys are saying the answer for this question is going to be Norman, Nandita, Muhammad, Zakib. Okay, C is what? C is going to be masculinization. Exactly. This is given in Human Health and Diseases. Anabolic steroids which are taken by the sport people sometimes. In the case of females, if at all they take the anabolic steroids, they will be having acne, they will be having mood swings, there will be fluctuation in their periods, and also they will be having masculinization and depression as well. So masculine body in the case of females is quite is not often and seen, and basically that is found only in the case of people who take anabolic steroids. Okay, so loss of memory, cirrhosis of liver, hallucination is not the one. The only answer for this question is going to be C. I hope that works. Yeah, let's go ahead with the next question. Works? Can you give me thumbs up if it's fine? Yes, Navya. Yes, Vishwanayak, Sri Ganesh, everyone. Perfect. Zainab. Guys, I'm so sorry I could not read out of most of your names from so long now, but I think that could speed up at least a bit in the class, right? So last question for the day. We save the entire forest to save the tiger. This approach of conservation is, we save the entire forest to save the tiger. This approach of conservation is in situ, ex situ, in vitro, in vivo. Lisha says A is the answer. Norman says last one. Okay, Panvit A, Preeti A, and Mohammed Sakib A. I want to read out everyone's name. Trisha A and Biredi A, Ramcharan A, Preeti, Sumaya, Kyun, Nandita, Mohammed Sakib, Sri Ganesh, Priyanka Patil, Norman, Kishan Gowda, Samarth, Arif, and Tarun, Iron Tom, and Vidya, Sarveen, Muhammad Atif, Arshia, everyone, Asif, Sania, Deadpool, Biredi, Shivalinga. I hope I'm not missing out on any names. Guys, I think Raghavendra, yes, therapist. So the guy answer for this particular question, Bharti Reddy, everyone, okay? And if at all I'm still missing out on names, I'm so sorry. It's going to be in situ conservation. Why? Because the tiger is over here. Listen, my tiger will not be looking like tiger. I'm pretty sure of that. Okay, but the tiger is right over here. And it not even looks like a cat. And basically to save the tiger, I am saving the entire forest out there. So isn't it in situ conservation? Isn't it in situ conservation since I'm conserving a lot of organisms in their natural habitat? Yes. So the answer for this question is going to be A. So that's a good note to laugh, right? So it would be A. Now, so Yes. Sagar, Ganesh and Vinod, Hema. Yes, perfect. So, ma'am, so beautiful tiger. Yeah. So, guys, this is it. These are the 60 questions which are there. And if at all you're wondering from where it has been taken, it's been taken from ABHYAS. If you feel, I'm not saying that you have to take it, if you feel that it is needed for you, if you want mock test, there are no too many full-length mock tests. There are basically part test and full-length test. Part test would be more. Part test means some chapters will be taken up, 60 questions will be made out of it. That is what is known as part test. Full-length means the complete syllabus will be there. So if at all you want to purchase, you can look out for it. The pricing for this is rupees 999. The link will be given in the description section. You can check it out if you wish to have it. Okay. That's your choice. Okay, it's totally your choice. And the next thing that is there, everyone who have attended this class and feels that it's good, the class was really good, please understand that we all teachers took a 15-day challenge, okay, series which was there. You guys might have attended, at least some of you, and there we basically took some chapters and we solved the PYQs. PYQs was the most important thing out there. Apart from that, extra questions were being solved. If you feel that you need help with the PYQs, or basically you should be, you have no idea how to solve the PYQs out there, you probably can refer to the PDFs of those particular videos. Again, it will be there in the WhatsApp channel. To join the WhatsApp channel, the link is there in the description section. You can just check it out. Okay. At least the PDF, if not the session, because I know you do not have time to watch the entire session out there. If you're watching any teacher session, you can watch in 2x speed also. But the PDF you can refer to for finding the questions and answer. If you have doubt, you can refer to the video. That is 15 days challenge. Now we are doing rank booster series, where what are we doing? Where we have completed one PYP, the whole paper of the previous year, 2025. And now this is a mock test paper that I am doing. Apart from it, we'll come with what? Complete revision also, chapter, subject by subject. My subject is there on Saturday. Cool. So guys, the reason I'm telling you about all this thing is that you have resources. Please check out the resources out there. Don't think that you do not have the resources. Okay. Oh my god, that was a fight. I'm taking a screenshot of that. Deadpool says, if you had gone a little longer, you could have matched Dhurandhar. Yes. So, children, ma'am, can you give the PDF of this? Yes, I can. Everyone, please join the WhatsApp channel. How to join the WhatsApp channel? I'll just put the link copied to clipboard. Everyone, subscribe to the channel also is what I'm hoping for. Yeah. And the link to the WhatsApp channel. Yeah, I have put the link to the WhatsApp channel. You can check out that. And ma'am, take rest right now. Yes, definitely. You also take rest. And Boas effect, you're asking me chemistry, ma'am. You go no. You're my god of biology. Oh my god. Yes. Nyampate, bye bye. Iron, bye bye. Please take care of yourself. And Arif, thank you so much. And how many marks are going to come from 11? So, Baba, what trend we have seen is out of 60 questions, 20 is the maximum number of questions that have come from 11th. Rest 40 comes from PU2. Okay, so yes, if at all you're worried about how to complete PU1, if you do not have much time, at least that PYQs would work, but you have to solve each and every PYQs religiously. Okay, yes. Okay, thank you so much, guys. We'll meet you really soon, and please take care of yourself. And there is a live today by Ankit sir for the complete math. If at all you want to watch it, you can watch it. And please explain question number 5. Please take care of yourself or your beauty fates. Oh my god, yes, I can explain question number 5. Hema, by the way, if you have any doubts, you can stay back. Okay, question number 5. 5. We have to go for 5. Yeah, I knew that there could be doubts in this. Okay, how are transformants selected from non-transformants? This is a question that we have, right? Thank you. Bye, Sunil Rathod. Bye, everyone who's saying thank you. Okay, so listen. Actually, the question is a bit twisted. It should have been recombinant and non-recombinants, but anyways, I'll explain. Okay, So listen to me. Normally, cells which are there, they do not have the plasmid. Okay, they do not have the plasmid. For example, I'm talking about PBR 322. This plasmid is not there. If this plasmid is there, what is going to happen? Whichever organism has this plasmid can survive in a medium that has ampicillin. Ampicillin is an antibiotic. Okay, so it will be able to survive. Why? Because it has a resistant gene onto it. Similarly, it will be able to survive in a medium that is having tetracycline, another antibiotic. Why? Because it has a gene into it. Right. Now, suppose basically I am making this particular plasmid out there and I'm letting the organism take this up. How will I understand whether the organism has taken up the plasmid or not? How will I understand whether it is transformed or not? The first thing that I am going to do I am going to put it in an ampicillin medium. When I put it in ampicillin medium, I get to understand that this is not able to survive. Why? Baba, it does not have the plasmid. That means it does not have the resistant gene. What about this? This will be able to survive. Why? Because it has the plasmid and it has the ampicillin resistant gene. What about this? For this also, I will be able to understand that it has the plasmid and has the ampicillin resistant gene. This will also survive. Okay, so am I not able to find which one is transformed and which one is not? Yes, I am. Transformed one, the one which has taken up something new, that is transformed, right? So I'm able to understand which one is transformed with the help of what? With the help of the selectable markers, which are nothing but your antibiotic resistant gene out there. Cool. Now listen to me carefully. Among them, so the question should have been how are recombinants selected from non-recombinants. That is why I think there is a confusion with that. But anyways, now the second thing that we do is, okay, they have taken up the plasmid, but have they taken up the plasmid with my gene of interest? With my gene of interest? So basically over here, if at all you see, this is ampicillin resistant gene, this is tetracycline resistant gene. But in this particular plasmid, I have made some changes. Instead of tetracycline, I would be having what? I will be having my gene of interest. This is my gene of interest. So basically, to find which one has my gene of interest, I put it in next thing, that is a tetracycline media. In tetracycline media, I am able to survive. I am able to see that this is able to survive, and this one is not able to survive. Why? Because I have put my gene of interest and it's now inactivated. The tetracycline resistant part is now what? Inactivated out there. Since it is inactivated, am I not able to find which one has my gene of interest? Yes, I am able to find that also. And that is the question. How are transformants selected from non-transformant? First thing, presence of alien DNA in the vector DNA results into insertional inactivation of the selectable marker. Presence of the alien DNA into the vector DNA, isn't it inactivating the selectable marker, that is the tetracycline resistant gene? Yes, it is. So this is one way. Second, antibiotic resistant gene gets inactivated. The same thing in different format has been put. Antibiotic resistant gene, my dear children, isn't it inactivated? Yes, that is also inactivated. Selectable markers can be anything. It can be antibiotic resistant gene such as tetracycline, or it can be a LAGZ gene which produces beta galactosidase and things like that. So it can be either of them. Selectable markers can be this, or it can be anything else, which is why sometimes it is just known as selectable marker without telling the actual name of it. So both these things are correct. I agree that the question should have been recombinants. It would make more sense. Is it okay? Are you able to understand it now? Are you able to understand? So I think yeah, so the person who asked me the fifth question, I hope, yeah, so where are you? Hema. Understood. Okay, perfect. Thank you so much. Guys, if there is no other doubt and 75% I can write the improvement exam or what? I didn't get your question. Sorry, dear. Okay, Bharti says 14th question. 14th question. Okay. The slurry left after methane production from the cattle dung is slurry left after methane production. Listen to me. Over here, you put the dung, okay? You put the dung, that is the cow dung, and you mix it with water. It goes into this particular area where, my dear children, in the cow dung, what do you have? You have methanobacterium, a bacteria, okay? This bacteria, what does it do? It produces biogas when it is having anaerobic environment. That means when there is no air. So it does that. It produces biogas. Now what is left behind? Can I use this as fertilizer and manure? Yes, I can use this because it is nothing but the cow dung itself, right? So that is being asked. The slurry left after methane production from the cattle dung is used as what? Is basically used as fertilizer. You should not be burning it. There's no point of burning it. If at all you can use it as fertilizer, it will be really good. Works, right? Mahesh, ma'am. Yes, ma'am, you're my favorite. Thank you so much. And Trisha, hats off to you, JJ, ma'am. Thanks to you so much. Okay. Yes. Bohr is not something that I can read off quickly by dear, but I have to check, dear. I do not, in PU1, if at all it's there. Just a second, okay? I'll just check. Ah, okay. Sorry. It will be there in that, what do you say, in the formation of oxyhemoglobin and carboxyhemoglobin, that part. Yes, it's there. Yes, I'm so sorry. So, guys, I think I'll end the live because I'm super tired. I cannot even stand now. Yes. Please make sure you watch through the entire video, and if you have any doubts, I'll be coming live again in very few days, yeah, and you can ask me all the doubts. Yes, Bohr effect is equals to more CO2, less O2. Exactly. Hemoglobin. My brain was not braining. Okay. Okay. Guys, so bye-bye. Please take care of yourself and please join the WhatsApp channel, and do not forget to hit the like button if at all this video helped you, and do not forget to hit the subscribe button as well. Bye-bye. Lots of love. Take care.